UP. She has more than 27 years of teaching and research experience. Her research interests include nanotechnology and drug targeting, as well as technology policy interventions for healthcare. She has 165 research papers published in journals of high repute and has authored 7 books and 22 chapters. She has also filed 2 Indian patents. Earlier, she was working as a professor in the Department of Pharmaceutical Sciences and director at Babasaheb Bhimrao Ambedkar University, Lucknow. She also served in various administrative positions at Central University, Lucknow. It's a pleasure to have you here, ma'am. Let's give her a very warm welcome. Thank you.
Thank you very much for the kind introduction. लग रहा है कि संस्थापक हैं उनको बहुत साधुवाद और बहुत सारे ऐसे तथ्य जो शायद इस दुनिया के लिए गुम जाते अगर कोई उसको आगे नहीं ले जाता, वो हो रहा है इस विश्वविद्यालय में। तो ये बहुत ही अच्छा काम है, अच्छा कार्य है और इसमें जहाँ हम जिस भी रूप से मदद कर पाएँ, we are always there. So I think I will be using mixed mediums while I talk, because many of you are Ayurveda students, and I would like to introduce nanotechnology to you, and to also inculcate a little interest in nanotechnology and how it can be used for herbal constituents and maybe Ayurveda also. Probably you're already working in this field, but I will share some of my work and some of the basic concepts for the students. Of course, it is not for the people sitting in the first few rows who are already into this deeply and they may not benefit from it, but the students at the back, this is, many of it is for you. So, talking about reaping the benefits of ethnopharmacology through nanoformulations, from nutraceuticals to pharmaceuticals, how we can utilize this. So you have this plant to patient series, which I think is a wonderful concept. So naturally, prevention is better than cure. And the first thing that you can do is to have a very healthy nutrition and a good lifestyle, and because of which you may not need medicines at all. That's what we are supposed to do, basically. So for that, I would think that the more colored substances that you are able to include in your diet every day, and what I say is try to take in 5 colors per day. So it could be red, orange, purple, the various greens. So those are the colors which if you take, you may probably not need nutraceuticals or pharmaceuticals at all. The next best thing that you can do is to take nutraceuticals which prevent disease. And then of course, if the illness or disease is already there, or if you are moving towards the illness, then you have your pharmaceuticals. All 3 of these can utilize nanotechnology. What I mostly work with is lipid-based nanotechnology. Why I work with lipid-based nanotechnology is that there are many lipids already available which have a lot of good in them. So if we take things like almond oil, or we take a lot of these antioxidant oils which are even there in fruits and nuts and seeds, if you are able to utilize them instead of a synthetic excipient, naturally it adds value to whatever formulation you are making. Also, every time that you include one new synthetic entity into the environment, there could be a possible hazard which may exist. So whatever exists in nature, if we are able to take a cue from nature and utilize it even to deliver a drug, it can be wonderful. So what I normally work with is lipid-based nanotechnology, microemulsions, nanoemulsions, NLCs, SLNs, nanoliposomes, etc. So normally, what are the factors that determine the bioavailability of nutraceuticals and other bioactive molecules? you have a lot of physiological factors which create a problem. Problem could be the first pass effect, it could be a high gastric emptying rate, it could be the effect of food, it could also be the restriction of the various barriers that exist in the body, it could be the enzymatic degradation of bioactives. So whatever you are taking as food, the quantity of the good part of it could lessen in its effect because of one of these things that is happening. So what we need to do is make some formulation in such a manner that you can maximize upon the effect of the nutraceuticals that are there. So by way of nanotechnology, we are now able to make healthier foods which have lower fats, lower salt, but desirable sensory properties. So talking about ice cream, I think कि जो students पीछे बैठे हैं उनको जरूर इस बात पे interest होगा कि आजकल की जो ice cream है वो कैसे creamier बनी, वो nano technology है. उसमें क्या किया जाता है, जो icicles होते हैं, जो पानी के बर्फ के छोटे छोटे कण हैं, उनको oil से coat कर दिया है. तो ये नैनो कण बन गए और इनको ऑयल से कोट कर दिया, इसलिए अब ये एक दूसरे से कोलिज नहीं हो रहे, ये बड़े पार्टिकल्स नहीं बन रहे. पहले आइसक्रीम में बड़े बड़े पार्टिकल्स होते थे जो आपके मुंह में आते थे और अब आइसक्रीम्स बहुत क्रीमी हो गई हैं. तो इसमें क्या हुआ, बहुत छोटी सी मात्रा में ऑयल लिया गया और जिसको कि बर्फ़ की छोटी सी आइसिकल पे कवर कर दिया गया और अब ये उतनी ग्रिटी फील नहीं करते और क्रीमियर फील करते हैं. सो दिस शुगर एंड फैट कोटिंग ऑन आइस क्रिस्टल्स हैड मेड द डिफरेंस टू आइसक्रीम and it has made it healthier and creamier because now the content of sugar, fat, everything is less because there's just a nanoparticle, a very thin layer that is available. So that is how nanotechnology in ice creams. Moving on, going green. So we can make nanoparticles of food grade materials which can be included in the various snacks that are available. And they could be proteins, polysaccharides, lipids, minerals, surfactants, which can be fabricated from nature. Now moving on to the lipid-based drug delivery systems on which I work. So over here, what do you have? What is the good part about it is that you have a core and a coat. Core में आप load करते हैं जो भी essential ingredient आपको load करना है. ये herbal origin का ingredient हो सकता है, आयुर्वेद से लिए गई कोई चीज़ हो सकती है. और इसके कारण आपको क्या मिलता है, आप उसको सरफेस पे डेकोरेट कर सकते हैं, इस तरीके से डेकोरेट कर सकते हैं कि ये कहाँ जाएगा और कैसे अब्जॉर्ब होगा, हम उसकी कोटिंग से डिसाइड कर सकते हैं कि ये कहाँ जाएगा और कहाँ पे अब्जॉर्ब होगा. तो इस कारण एनहांस्ड ब्लड सर्कुलेशन हो जाता है, पैसिव टारगेटिंग हो जाती है कि जिस चीज़ को हम जहाँ पहुँचाना चाहते हैं, अगर वो नैनो पार्टिकल है तो वो अपने आप वहाँ पहुँच जाएगा. अगर हम उसे एक्टिवली कोट करते हैं तो हम जहाँ भेजना चाहते हैं, हो सकता है एक ट्यूमर हो सकता है कि कोई इन्फ्लेमेटरी डिजीज़ हो, जहाँ इन्फ्लेमेशन हो, हम वहाँ उसको भेज सकते हैं कोटिंग के द्वारा. सो ओवर हियर यू सी दैट साइज डज मैटर. अगर कोई बहुत ही छोटा फॉर्मुलेशन है, क्योंकि वो छोटा है तो वो अपने आप एक सेल के अंदर तक पहुँच पाएगा, जो कि पहले डायग्राम में आप देख रहे हैं. So that is passive targeting only by virtue of size. The second one, you can make it long circulating nano carrier. मतलब उसके ऊपर एक ऐसी कोटिंग करें कि उसको सेल का जो रेटिकुलो एंडोथेलियल सिस्टम है वो अपटेक ना करे, उसको ले और उसकी कोटिंग के वजह से ये एक बार, दूसरी बार, तीसरी बार, बार बार जब सर्कुलेटरी सिस्टम में गोल घूम रहा है तो चांसेस हैं कि ये ज़्यादा से ज़्यादा अब्जॉर्ब हो जाएगा. सो दिस इज़ समथिंग विच हेल्प्स बिकॉज ऑफ द कोटिंग एंड मेकिंग इट स्टेल्थ. जैसे कि पुलिस वाले एक यूनिफॉर्म हरा और ब्राउन कलर का पहनते हैं कि पत्तियों में उनको छुपाया जा सके, वैसे ही नैनो पार्टिकल्स को इस तरीके से कोट कर दिया जाता है कि वो स्टेल्थ हो जाते हैं. बॉडी का रेटिकुलो एंडोथेलियल सिस्टम उनको पहचानता नहीं है और इसलिए वो बार बार सर्कुलेट होते हैं और ज़्यादा से ज़्यादा मात्रा में अब्जॉर्ब हो पाते हैं. द थर्ड थिंग दैट वी कैन डू इज़ एक्टिव टारगेटिंग, where we put something or the other on the nanoparticles so that it is able to go effectively to the required site. So over here we make use of many different types of strategies for active targeting. So the techniques for nanoparticles, normally nanoparticles सुन के ऐसे लगता है कि ये सारी किताबी बातें हैं और शायद ये possible नहीं है, but it is very much possible, and there are a lot of techniques that are available these days which you can also make at a large scale. So the modern techniques that we have are things like a microfluidizer, which are very thin channels, and because of the high impact of the liquid that is flowing, we are able to make nanoparticles with them. You have ultrasonic homogenizers, you have various techniques which are maybe chemical in nature also, like you have emulsion, inversion techniques, etc. You have a high pressure homogenizer, which is only pressure that is being used. So if you are able to reduce the particle size only with the help of pressure, you are not using any other method, and that is why you make it relatively safe and good for use. So there are a lot of things which already exist in nature and which are being utilized, and the active ingredients are normally taken because let us say that if you say haldi तो हम रोज अपने डाइट में लेते हैं, but अगर हल्दी को एंटी कैंसर पोटेंशियल के लिए यूज़ करना है तो हो सकता है कि आपको आधी ग्लास हल्दी लेनी पड़े, जिससे कि वो टारगेट ऑर्गन जहाँ तक आप ट्यूमर में भेजना चाह रहे हैं वहाँ तक जाने के लिए बहुत ज़्यादा क्वांटिटी चाहिए. तो फिर क्या होता है, इसका जो भी एक्टिव इंग्रेडिएंट है आप वो लेते हैं, उसको आप नैनो फॉर्मुलेशन में यूटिलाइज करते हैं, उसको इस तरीके से कोट करते हैं कि आप कौन से ऑर्गन में, हो सकता है ब्रेस्ट है, हो सकता है स्किन है, कौन से ऑर्गन में कहाँ तक पहुँचाना चाह रहे हैं आप उसकी कोशिश करते हैं. तो नेचर में ऐसे बहुत सी चीज़ें ऑलरेडी अवेलेबल हैं. पतंजलि इस पर बहुत अच्छा काम कर रहा है, but what I am telling you is just to explain it to the students at the back that definitely there is a lot of potential and these are the finer points. So this is like a Trojan horse concept, जैसे कि काट के घोड़े में लोग अंदर बैठ के और चले गए और किसी को पता नहीं चला, उनको लगा काट का घोड़ा आया पर इसके अंदर बहुत सारा कुछ था. ऐसे ही जो मेडिकमेंट्स हैं जिनको कि भेजना होता है शरीर में, उन्हें ऐसे ही भेजा जाता है नैनो पार्टिकल्स द्वारा कि वो प्रोटेक्ट होके अंदर पहुंच जाते हैं टू द ऑर्गन और द सेल वेयर यू वांट टू मेक इट रीच. सो व्हाई नैनो? Nano because nano is the scale on which the biology, the physics and the chemistry they come together. तीनों चीज़ें वहाँ पे इस तरीके से हो रही होती हैं that you can maximize the effect of nanotechnology. That is when you require nanotechnology. So let us say enzymes. Enzymes are something which degrade very rapidly, and that is why there are many diseases that happen when the right enzyme is not present at the right place. So what can be done is, in nanoparticles, you can either immobilize those enzymes or you can have them in a form of a bioconjugate so that this conjugate breaks only when you require it, and these enzymes don't get released prematurely. So that is how enzymes are used for therapy. There are a lot of enzymes like urease. So you may have heard that there are children who are allergic to milk. And naturally, if milk is one ingredient in India which is used in so many things, मिठाई में, ice creams में और chocolates में, बच्चों को बहुत ही ज़्यादा पसंद है. तो अगर ऐसा बच्चा जो कि मिल्क से एलर्जिक है, उसके लिए उसकी ज़िंदगी बदल जाती है और उसके आसपास के जो भी उसके फ्रेंड्स हैं वो कुछ और खा रहे होते हैं और उसको कुछ और खाने को मिलता है. इन दैट केस व्हाट कैन बी डन इज, ये मिल्क एलर्जी के लिए कौन सा एंजाइम रिस्पॉन्सिबल है जो आपको चाहिए जिससे कि ये एलर्जी ना कॉज करे और ब्रेक डाउन कर ले, उसको ऐसे मिल्क प्रोडक्ट में उसके साथ दिया जाता है इमोबिलाइज करके बाय विद द यूज ऑफ नैनो टेक्नोलॉजी. सो डेफिनेटली दिस कैन बी डन. So now coming to some research work that my group has done in the past. So you know about resveratrol. Resveratrol is something, an antioxidant which is present in grapes. काले अंगूर जो हैं और ऐसे colored berries हैं उनमें present है. तो हमने इसको लेके और नैनो पार्टिकल्स बनाए और उसकी ब्रेन टारगेटिंग की, तो इफ़ेक्टिवली बहुत सी न्यूरो डीजेनरेटिव डिजीज़ के लिए ये काम आया. रसभरी आजकल ये फ्रूट मिल रहा है. द ऑयल दैट वाज अवेलेबल फ्रॉम दीज केप गूसबेरी ऑयल is something which we used when we made lipid nanoparticles. The lipid part of it was the oil of Physalis peruviana, or raspberry as most of us know it. And this is full of antioxidants. So when we utilized this oil to make our nanoparticles, we were able to do quite a lot with this. So probably our next generation is going to be like Jessica, who's 2 years old, and she can detect nanoparticles of zucchini or lauki or whatever you may name it, even in poor light, because that is how food is going to be in future. So coming to chronic inflammatory diseases, you know that these are diseases which people take lifelong. They take painkillers and non-steroidal anti-inflammatory drugs because, and that dose keeps increasing because that threshold keeps increasing. So what we want is, how can we do something about this, that so much of NSAIDs does not have to be taken. So because of this, we explored thymoquinone. Thymoquinone is a drug which is a bioactive phenolic compound and which is present in the black seeds of Nigella sativa, which is kalonji. So we took this bioactive thymoquinone and we decided to explore it, the properties of thymoquinone, including the anti-inflammatory, the antioxidant properties, including the down-regulation of anti-inflammatory cytokines like IL-beta, TNF-alpha, PGE2, and up-regulation of anti-inflammatory cytokines like IL-10, etc. So the problem with this was that it had poor aqueous solubility, and that is why we decided to make nanocarriers out of it. So this is a brief about the experiment that we carried out, the methodology that we carried out. And then finally we went up to animal studies and we saw the effect of the nano formulation that we made. So over here we took tamanu oil. Tamanu oil is again an oil of, it is a naturally occurring oil, and it has proven anti-inflammatory and antioxidant properties. And in this we loaded our thymoquinone, which I just told you about. And with the help of this and a few synthetic lipids as well, like stearic acid, glyceryl distearate. So these are basically of plant origin, but they are made into something else. So they are like semi-synthetic in origin. So we took these and then we made our nano formulations out of them. And the results that we got were quite good. I'm not going into details because 20 minutes is the time that's been given to me. So of course we designed the formulation through box-by-Henkin design. We optimized the ratios of the various ingredients that we wanted. And the optimized formulation, we went ahead with many other studies like XRD, the morphological characterization through FESEM. Over here you can see the formulation that we made. Then we went in for ex vivo skin permeation studies of this formulation. And we found that there is increased permeation due to the presence of fatty acids in the skin and tamanu oil, which aids the solubility and the permeability of the entrapped thymoquinone, which is what we wanted to do. We went in for in vitro cell cytotoxicity studies, including MTT assay, and then in vivo studies where we went for a carrageenan-induced model. And this therapy lasted for 8 days for arthritis induction, and then we followed up with the treatment up to day 27, and then we did the further studies. We also did it on another model. So FCA-induced rheumatoid arthritis model, we also did on that. These were the arthritic scores. First you can see swelling and redness, and after that you can see that there is improvement in the arthritis. So we went for radiography, and these are the results of the X-ray images that we took. And initially you can see a gap in between the bones because of the swelling that is there, and then the gap is lessened. So that was what this study was about. We also did cytokine estimation in serum using multiplex plate reader system. And to sum up, the current study explored the pharmaceutical importance of tamanu oil as an oil in the drug delivery system and the natural bioactive thymoquinone. So you can see that it makes a lot of sense to take cues from nature. Nature के पास बहुत सारे answers हैं और हमें उसको बस explore करने की ज़रूरत है. A few of my publications, synthesis of pH-sensitive cross-linked guar gum, polyacrylic acid, acrylonitrile, co-acrylonitrile for the delivery of thymoquinone against inflammation. This was another experiment that we did, not the one that I've just discussed. This one of the papers. Then over here you can see that we did hyaluronic acid functionalized PCL nanoparticles in urethane-induced lung cancer. We took biotin anchored NLCs for targeted delivery of doxorubicin in the management of mammary gland carcinoma. We also took naringenin, another natural ingredient, and then we investigated that for microemulsion loaded sericin against UVB-induced photoaging. We got good results. We also took tamanu oil potentiated novel sericin emulsion. Sericin is a protein which is found in silk, and it is a byproduct while making silk. So you can see that there are a number of natural ingredients which can actually be utilized and where you can lessen the load on the environment by eliminating as many synthetic agents as possible and including as many natural agents as possible. So this was another experiment that we did with inositol hexophosphate for its anti-cancer activity. Inositol or IP6 is an ingredient which is present in normal pulses. Dal jo hum lete hain usme hai, and normally it's regarded something bad because it's supposed to be an anti-nutrient. But when you make it into a nanoparticle and when you're able to target it to a tumor, it has shown remarkable potential. This was again another type of nanoparticle we made with IP6. Over here we use jacqueline. Jacqueline for coating. Jacqueline is a substance which is present in jackfruit. So you can see that time and again we have been using so many natural ingredients. This is the co-therapy of capsaicin, जो कि मिर्च में, शिमला मिर्च में और मिर्च में पाया जाता है, उसको कैसे यूज़ किया एंटी कैंसर थेरेपी के लिए. तो these are a few of my papers, and to sum it up, controlled and sustained release through nanoparticles is very much possible using natural ingredients. Lipid nanocarriers are very safe delivery systems because we use lipids which are normally present in the environment, and our body has lipase, 5 different types of lipase by which we can break them down so it makes it green. That is possible. And nano capsules and nano objects for an on-demand preservative, enriched food, flavor, smell, taste, color, all this is possible with the help of nanotechnology. So thank you very much. Acknowledgements to my research group, ACT BBAU, which was my previous organization, and Naiper Raebareli, which is my present organization. And I'm open to questions, if any.
Ma'am, thank you very much for a fantastic presentation. We have you for first time, and we are very happy that you are here and sharing all the information that you have. We really get mesmerized by so many nanoparticles that you have generated, and specifically the one you talked about, tamanu oil. So I want to just understand what is this oil and where this is coming from, what the source of it.
Okay.
And my second question is that you talked about nano emulsion, nano encapsulation has helped permeation of the particles. But have you tried any way to permeate the blood-brain barrier? Can we actually push the compounds beyond that barrier so that we can have a delivery right into the brain or in CNS? So your insights on those would be very helpful, ma'am. Thank you.
Yeah. So then, the first question you asked about was tamanu oil. Tamanu oil, the name is tamanu oil, although it sounds like it must be having some English name, but it's called tamanu oil. And there are tamanu nuts which are available. So this tamanu oil is already available just as the oil. And although an extraction and all that can also be done from the tamanu nuts, but it's also freely available as tamanu oil itself in the market.
So did you isolate oil yourself,
no,
or you bought a commercial one?
No, we bought it commercially. We just ensured that it was pure tamanu oil, and we worked with it. Because over here it was just one excipient in our formulation. So we did not want to prolongate the work so much that we go first for extraction and all that. So that we just bought from the market. So that was about tamanu oil. The second thing you said was about blood-brain barrier. So there are various routes of drug delivery also which are available for blood-brain barrier. In today's presentation, I have not talked about it, but then very long ago, we did do proof of concept of nose-to-brain delivery, where you need to push the formulation of the nanoparticles deep inside the nose. And from there, there is one place where it will not go into the circulation, it goes directly into the brain. So as long as when you're taking nano formulation, you are taking a very potent medicament because you cannot load a lot into a very small nanoparticle. So something which is very potent, you can take, and then you have to put a very small quantity inside, and that is possible. Otherwise, more than that, patient compliance will not be possible. So in this case, patient compliance is possible when it's a very minute quantity that you're putting, and it is a direct movement from nose to brain. So that was one way of doing it, where you can take that. And the other thing for brain delivery, we also utilized astaxanthin, which is supposed to be a brain antioxidant. And it is also naturally available in nature. So we worked with astaxanthin because we knew that it will automatically go directly into the brain. Otherwise, there are other ways where you can temporarily make the tight junctions a bit loose, but then it only happens temporarily, or it happens in cases of tumor, etc., where because of the pathophysiology of the disease, for a certain period of time, that tight junction is loose. And so, till the time that the disease reverses, the tight junction is loose and the movement through the blood-brain barrier is more. So those are some techniques which we have utilized in the past. Not that I've done a lot of work on it, but yes, some work definitely.
Just to continue on this, has someone really done a tissue distribution experiment on a compound which is very polar in nature, get coated with some sort of oil or solid lipid nanoparticle form of that compound, and does it really get compartmentalized into CNS or in the brain?
Okay, so this, the deep nose, the nose to brain delivery that I'm talking about, we went ahead and did the entire thing. We targeted, we tagged it with technetium, and then we saw the movement, and then a lot of it was concentrated in the brain. So we got good results with nose to brain delivery, really good results with that. So that was through a solid lipid nanoparticle that we were able to get nose to brain delivery.
Thank you, ma'am. So kind of you. Thank you.
नमस्कार मैम।
नमस्कार।
ये तमानू ऑयल जो है इसका बोटेनिकल सोर्स है कैलोफिलम इन्नोफिलम
जी,
एक ट्री है, तो उसको लोकल भाषा में तमानू कहते हैं
जी,
उससे ये एक्सट्रैक्ट किया जाता है।
हाँ हाँ
जी जी,
yeah. So what I said was it's available as tamanu oil in the market. You don't have to worry, like it's not some vernacular name that I'm using. It's available as tamanu oil.
थैंक यू।
Thank you so much, ma'am, for giving your insights on reaping the benefits.
Hello, excuse me, sir. One more question, please. Yeah, with the permission of His Highness Swami Acharya-ji, and I have a small question, madam. We have young doctors practicing, and we have the ancient era of children, but now we are entering into a new era of nanoparticles. Do you suggest any specific equipment for making nanoparticles at the clinic level? And its brand, because we may ask our Highness Acharya-ji to buy for the institute, and also make our young doctors to make the nanoparticles of the children for the practice. Thank you.
Sir, just to interrupt, those equipment are available at PRI. Whenever you want to come and see, you can easily come, see and use as per the requirement of yours, student and yours as well, sir. So please feel free.
Thank you so much. Thank you so much.
I hijacked your answer.
I know. There are so many hidden things in PRI which we have to know a lot.
Thank you so much, ma'am. I would now request our next eminent speaker to deliver his talk on neuroprotective mechanism and Ras Sindoor by Professor Ganesh ji, sir. Professor Ganesh is presently the Senior Professor in Department of Biological Sciences and Bioengineering and also Deputy Director at Indian Institute of Technology, Kanpur. Dr. Ganesh is known for his contributions in dissecting genetic pathways of neurodegeneration and his attempts in therapeutic interventions for neurodegenerative disorders. We are honored to have you here, sir.
Namaskar. Thank you very much for the kind introduction, and my apologies that I could not be there physically. I planned to be there, but for some unknown reason, the trains got delayed, so I couldn't be there, but I would certainly visit the place in the near future to have interaction with Patanjali. So we had some interaction for a collaborative work between our two institutions. That is something that I am very, very keen that we should take it forward. Just to give a brief idea about the kind of work that we do in our group at IIT Kanpur is that one of the major projects that I work on is on epilepsy. One is a familial form of epilepsy called Lafora epilepsy because it's a genetic form. And then I also work on glycogen and its role in neuronal stress response because glycogen normally thought to be a bad biomolecule in the neuron. So we try to understand what exactly the function of the glycogen and why in certain conditions you have the glycogen accumulation. And of course, we dissect the pathways that lead to the death of the neurons in the brain. As a result, you have neurodegeneration, either because of a disease like Alzheimer's and Parkinson's, dementia, and so on, or as part of normal aging that we see. And of course, when trying to, even now, recently we looked at some projects to look at how the parental age can influence how the neurodevelopmental events, including susceptibility to the disorders when as the person ages. Just to give a summary of what I'm going to speak today is about the neuroprotective mechanisms of Ayurvedic formulations, Amalaki Rasayana and Ras Sindoor. And this work was primarily done by Bhupender Verma, one of my former PhD student, and it was started by Aditi Gupta when she started her MS master's project, and Priyanka helped both of them. And I would like to place on records, because my introduction to the Ayurvedic biology, especially looking at how the formulations function, what's the molecular mechanism, is something that I would like to thank these two experts, Professor Valiathan and Professor Lakotia, because they asked me to really investigate this process, and that's the reason why I've gone into this particular topic, and this has become now one of my major verticals in the group. And also I would like to acknowledge the funding from ACRB and IIT Kanpur for this event, for this particular project, right? So just to look, give a brief overview on what are, neurodegenerative disorders. I'm sure many of you are there in the audience know quite well that one of the common features of neurodegenerative disorders is that inclusions that are seen in the neuron. This could be protein inclusions like what you see in polyglutamine disorders like Huntington, or in Alzheimer's, or in ALS, like you have inclusions, and of course the very structural property of the inclusions, protein inclusions in prion disease, which sort of having a cascading effect, is also well known. But what my group primarily looks at is the inclusions that are rich in glycogen, and it is very prevalent. It is not something restricted to a few diseases. It is seen in multiple disorders. Not many groups have really worked on it, but that is something that one should look at because it's also seen in aged brain. That could be one of the reasons why the neurons or the dementia sets in. If you really look at the current understanding on how neurons with age or in certain disorders, how it sort of gives up its functionality is because of the stress that it experiences, right? So the neurons live as long as we live and they don't divide. So any accumulation there is going to affect the way the neurons live or their function, and that would manifest as our cognitive function or even our other activities that are directly controlled by the brain. So if you look at carefully, there are a number of mechanisms that are affected by the stress. It could be oxidative stress, it could be any other stress that affects the cellular process. And then of course you have several players which kind of mount a response, which is called as stress response pathway. Like one well-known mechanism is the ER stress response, or for example, heat shock response. It protects the cell from such kind of an insult. But if the stress is persistent and it could saturate your response mechanism, then slowly the neurons die. And that is what the intensity of the stress. If it is like a mutant gene that keeps making an abnormal protein, then the stress increases with time as a function of age. Or if a gene is mutated and its protein is involved in stress response pathway, due to the mutation, then the stress response pathway is compromised, and you have the neurons that have the deficiency, therefore even a normal load of stress they cannot handle. So that's the background, but as you see in aging is that there are a number of processes that that are altered, and you can see that, for example, you have abnormal protein assemblies, you also have protein aggregates that come as part of the aging, and same kind of structural changes, pathological changes, you see it even in neurodegenerative condition as well as in the aging. So it looks like the processes are similar, maybe the trigger could be different. One is earlier onset, other one is late onset. So just to give an idea how this, I've modeled this particular disease. It's a genetic form of disorder. The age at onset where you see the symptoms is 12 to 16 years, and the patient die within 10 years of onset. That's one of the very fatal form of disease. It's very prevalent in the southern part of the country. There are a number of families, there are about 1,000+ families that are known already affected with the disease. And the defining symptoms, of course, seizures, and these are mostly myoclonic seizures, but they also the patient develop ataxia, they're unable to walk, and then they have a dementia within, 10 years after the onset. So even they forget what they have learned 5 years back. And of course, they have a psychosis develops. And if you look at the organs and tissues and cells, then you would find these kind of inclusion that you see here. These are carbohydrate-rich inclusions. This is a EM picture of the inclusion that is present in a variety of tissues. So this is one common feature in this particular disease. So what we know is there are two different genes that are affected. I will not get into the details. One of the genes that codes for an ubiquitin ligase, the other one that codes for a phosphatase. So these two proteins basically modifies the target protein. So that's the major function. And if any one of that protein is compromised in its function because of a mutation, then the patient develops this deadly or fatal disease, right? And of course, these two, if you look at the families that have mutation in phosphatase or ubiquitin ligase, they have a clinically very similar, phenotype. Therefore, we would assume that both proteins function in a common pathway. So if you have defect in one, it is equivalent to not having the other two. So that's why you have very similar symptoms and prognosis. Much of our understanding on the disease have come from the models that have been created, one of which was created by me, which we have been extensively characterizing the last 2 decades, is the knockout mice, laforin, the phosphatase deficient mice. And if you look at about 1 year since the birth, the animals that lack this phosphatase, develop most of the symptoms that you see in the human. It has a neurodegeneration. It has these glycogen inclusions, what is called as Lafora bodies in the brain and in the other tissues, including liver and muscle. And they have impaired behavioral response very early in their life, for 4 or 5 months. And they have ataxia, and of course they also develop epilepsy, or they are more susceptible to epilepsy. I'll show you a little later as they age. That is what it is. Only difference is that they don't die early. That's the one difference between the human and mouse. Otherwise, they develop most of the symptoms. So our group has done a lot of work, and many of the papers that are shown here are published from here, which clearly, sort of established multiple roles for these two proteins in cellular context. So they play a significant key role in glycogen metabolism, in ER stress response, heat shock response, inflammatory response, mRNA processing, mitochondrial quality control, autophagy, and many of this may also contribute to how the neurons form their network and how do they maintain memory and so on. So that is dendritic spine dynamics. So, and that, pretty much offers a very good model to understand how rasayanas can protect the neurons and what's the mechanism. So we can validate, we can give a scientific proof that, our age-old traditions they have. So that exactly is what the talk is about. So I'm going to not say something new with regard to their function. It is known that they are neuroprotective from the human use, but what has not been dissected quite well is their molecular mechanism, and that has been our attempt to show that indeed they work, and this is the process that they work on. And these are the two formulations that we took, the Amalaki Rasayana and Sindoor. I'm sure many of you over there know a lot about these formulations than me. Of course, this first one is, of course, the Indian gooseberry, the formulation, Amalaki powder with honey and ghee. The other one is more of a, what do you call as the metal and combination, right? So that's what it is, Sindoor, sulfur and the aerial root of the banyan tree. So that's the formulation. This is again, this is not something that we have formulated. These are the formulation that is well known, and we use the same formulation to check how they function and how do they protect, what's the mechanism through which they protect the neurons. There are a few papers earlier that is mainly from Professor Lakotia's group, where they have characterized these two rasayanas using fly model for example, in Huntington disease. That has given some insight as to how that possibly functions, in the cellular context. So we have used a very well-defined genetic model for a neurodegenerative disorder to, show a lot more, pathways that are similar to the human system. So we have used two different mouse models for the same disease, Lafora disease. One is in a mouse deficient for the phosphatase, other one is mouse deficient for the ubiquitin ligase. So we have mixed these rasayanas with a jelly, gelatin, which is coated with chocolate for inducing the animal to eat, and that's this dosage that is shown at the left corner here. And these rasayanas we obtained it from the Kottakkal Arya Vaidyasaila. And they're well characterized formulations. And what we have done is we fed the animals, these animals from 2 months to 8 months. That's basically 6 months period we fed them. And then we looked at what happens to these animals with regard to the pathology they develop, or the symptoms they develop, or cognitive functions, and whether this formulation is able to delay, arrest, or restore any of the defect that otherwise that these animals express, develop. These are some of the things that we have looked at. You look at susceptibility to epileptic seizures by inducing epilepsy, and then behavioral assessment with regard to their cognitive abilities, and then you look at many of the molecular pathways that are known to be compromised in this animal model. So the way we have scored seizure is that we have injected a chemical, which is the PTZ, or pentylene tetrozole, which is known inducer of seizures in animal. and it is dose dependent. The wild type animals for a given dose will not develop a seizure, but if you increase the dosage, they will develop. But animals that are prone for seizures, even at a lower dose, they develop seizures. So that's the model. And this is a GABA receptor, antagonist, and it's well known in the literature that it would have seizures, and if the dosage increases or the animal is extremely vulnerable for the induced seizure, then they would die. That is the highest point of their seizure induction. Right? So this is just to show you that how the seizures are induced. Somehow I couldn't play the video here, maybe because of the Zoom platform, but what is known is this is the wild type and this is the deficient mice. We know that I'm unable to show the video, but when you inject and if you follow through for 5 minutes, then they develop conversions. Here the wild type wouldn't have shown any seizures, but the Laforin mice would develop various stages of seizures. It could be a freeze, or it could be the head nodding, or the whole body, a kind of, they climb up and they have the tonus, or it could be a tonic-clonic seizures wherein they lay flat and, and while running and jumping, and it can be very severe, it can even lead to death. So this is the different stage, and this is more milder stage, and this is much more severe stage, and then we can score for each of the stage and duration. We'll be able to plot a kind of severity. So that is what's shown here, seizure severity. You can see that the Laforin deficient or Malin deficient animals have very high seizure severity as compared to the wild type. And even the duration of seizures, if you look at it, is far more greater than the wild type. The question is what happens to animals that have been fed with these rasayanas? And you can see that for both models, when you feed the animal with the either amalek rasayana or sindoor, the seizure severity drastically reduces. It almost reduces the seizure severity, and even the duration is very much reduced. Clearly shows that this rasayana is able to sort of restore the normal function of the neurons. Therefore, they are no longer as susceptible as they are in animals that are not treated with these rasayanas, or the ones that are treated with the controlled jelly, right? So this is the other aspect of the inclusions. You can see, I don't know whether you could see here, there are pink dots that you see here, and these are the glycogen inclusions, but we did not find much of a difference in the inclusions in the animals that are treated with these rasayanas. So in other words, the glycogen metabolism or glycogen storage in the neuron that you see in the disease are not sort of affected or altered by the rasayanas. So whatever seizure susceptibility that is able to rescue could be independent of this glycogen. That is something that we'll see now. One aspect that is already known in this in the disorder is that the animals that are deficient for the phosphatase or the ubiquitin ligase are prone for neuroinflammation. You can see here, as compared to the wild type, these animals show higher reactivity for the antibody against, for example, the IBA1 or other, neuroinflammatory marker, and that is, something that we have looked at, for example, in different areas, and you can see that you have a higher reactivity for this marker that shows that neuroinflammation is sort of enhanced in these animals. And obviously, we wanted to see is that if you treat the animals with the rasayanas, whether it is able to suppress the neuroinflammation, and we do see a significant reduction in the neuroinflammation for some of the markers. It's not only in the immunoreactivity. We have also quantitative PCR where you can measure TFAP, IBA1, COX2, TNF-alpha. These are standard markers of neuroinflammation, and you can see it in majority of them. You would find that the rasayanas are able to bring down the neuroinflammation. Perhaps that is one reason why the seizure susceptibility has been restored because it is able to, prevent or delay the neuroinflammation, these two rasayanas. The other aspect that is well known in neurodegeneration is one of the response pathways. It's called autophagy. This process, what happens is that you have a membrane that forms. We believe that it could be the endoplasmic reticulum or nuclear membrane. Still, there is a lot of debate. But this membrane really forms a vesicle, and this vesicle, double membrane vesicle, that sort of takes away all these protein aggregations or damaged mitochondria, and then this vesicle fuses with what is called as lysosome to degrade the substance. Therefore, these abnormal forms of either protein or the organelle are destroyed within the neuron. Therefore, the neurons can live healthy. So this autophagy is extremely important for the neurons, and if neurons are compromised for the autophagic function, it leads to their death, neurodegeneration, and the disease. So this process is compromised, known to be compromised in the Lafora mouse model. I will not show the details, but when we have really looked at whether this process is sort of helped by the feeding the animals with the rasayanas, what we found is that indeed that's not the case. It doesn't really alter the autophagic process, so therefore it is something else. The rasayanas did not really change the autophagic process, but perhaps it is acting on some other pathway. One other pathway that is also compromised in this condition and also in many other neurodegenerative conditions is the heat shock response pathway. It is not only a heat shock response, because this pathway is a stress response pathway. It is activated in a variety of conditions, including, for example, hypoxia. And a master regulator for the heat shock response pathway is the transcription factor, what is called the HSO1 or heat shock factor 1, which goes and activates a large number of target proteins which are chaperones. And you will find in this animal, for example, in Laforin deficient mice or Lafora disease mice, the level of HSO1 as well as targets are low. So therefore, it is compromised both at the protein and transcript level. Therefore, any insult to the neuron, they cannot really respond to, and that's why the neurons die faster. And we have shown earlier that if we can restore the function of the shock response pathway, then you are able to suppress many of the symptoms in this animal. And that is something that we looked at, whether the rasayana help, and at least this amalaki rasayana restores, for example, HSP70 and HSP40 level. These are HSP1 targets, suggesting that they are able to bring back the homeostasis in terms of protein function. The other function that is compromised, as I told, we are not looking at the protein or the pathways. Now we are looking at the neuron as a whole, is how they formed the networks, how do they form the synaptic connections, and how that may help in the memory process itself. So that is called as dendritic spines. These are very mobile and plastic structure which either form and stabilize, and if there are no signals, then they will not be strengthened. That's one of the reasons why you have memory formation itself. And it's a very, very dynamic process, and it's again a lot of studies that have been done. And it is known in certain conditions, for example, in ASDs, the dendritic spine number increases, and then as a function of, for example, age, it comes down. Even in Alzheimer's disease, for example, the number of spines goes down with the age, and this is something that, as I told you, is a dynamic. Over the time, it changes, and it also depends on what kind of signal that we receive in terms of learning and ability. And it's not only the number, but the shape, really matter. What kind of shapes these spines have, that talks about the maturity of the spines. And in conditions like Fragile X, Rett syndrome, these are mental retardation where the IQ level of the individuals are low, you have more immature spines, very thin, projections. But in a typical condition, you will have this kind of, mushroom-like structure, which is considered to be the more mature form of the dendritic spines. And what you can see is that, it's known that in Lafora disease, the spine number increases, and mostly these are thin, immature spines. And what we have shown is that if you feed the animal with the rasayanas, either Amalaki rasayana or Rasindur, the number of spines come down, and also the density of the spine increases, saying that they get mature. So if that happens, does it increase the cognitive function? And that is one of the readouts I'm showing, that we can use what is called the novel object recognition test as one of the readouts for the cognitive function. Here what is done is that animals are exposed to a similar type of, a kind of a, it could be a ball, it could be some shape, a structure, a novel object. But then they spend time, they understand this, and then they lose interest. But after some time, if you replace one of them with a new subject, then they would spend more time on this subject than this, because they are already familiar with this particular object. So by looking at how much time they have spent between these two, when you are introducing this after some time, you will be able to test the memory level of these animals. And you can see here, for example, in the wild type, So this is the green one, that is the novel object. When you introduce this object second time, when there is a new object is given, the animals spend more time exploring this novel object. And you don't see that kind of a distinction for the Lafforin deficient mice, suggesting that they are unable to distinguish the novel versus already familiar object. That means they lack the memory, right? And so is the case with the Malin deficient mice. But if you treat either of these animals with the rasayanas, and certainly you see that they are able to again discriminate the novel object from the familiar object, saying that it is able to remember better. And perhaps this is because of the dendritic spines now mature as a result of this rasayanas. That's clearly is one readout. Of course, one need to really study a lot more. And this is only to give an idea as to how the rasayanas can alter the physiology, both at molecular level, at the process level, at the cellular level, to improve the cognitive functions and so on. So just to summarize my talk, so what I have shown is that loss of any one of these two proteins, the phosphatase Lafforin or the ubiquitin ligase malin, results in a condition which is fatal, which involves multiple symptoms, including myoclonic epilepsy and the abnormal accumulation of glycogen, compromised stress response pathway, and behavioral deficit, including cognitive abilities. And that happens because of either you have autophagy, compromised autophagy, or compromised heat shock response, or neuroinflammation, which may modulate the way the dendrites form, mature, and maintain. And when you have these, formulations, the animals are fed with formulation for reasonable long duration, it is able to restore or partially restore some of these pathways which help, for example, even the maturation of the dendritic spines, which sort of restores or partially restores some of the deficits that you see in the model. So that's my little brief work on the disease. We also are using very similar approach to understand how these formulations can modulate the aging process, whether that, whether the dementia can be arrested, whether the, other processes. That is something that we have been working on. And maybe with this, I would end my talk, and I'll be very, very happy to take any questions, and I look forward to the questions and comments. Thank you.
Can I have a question there? Am I audible to you, sir?
Am I audible to you, sir?
Yes, yeah, I could.
So, sir, this is Anurag here. Thank you very much for this presentation. I have just two queries. That you generated some fancy jellies as a food for the animals. So how did you make sure that their nutritional requirement is taken care of? Because you would have to feed them for close to 6 months. And my second question is that you had a
Sorry,
fantastic...
it's not audible enough. If someone could, in the podium, can restate that question, that would help me.
Wait a second, sir. He's coming to the podium just right now.
Thank you very much. Yeah, I appreciate it.
Hello, sir. Hi, this is Anurag here. Thank you very much for this presentation, and good to see you on a virtual screen. We hope to see you real sometime this month, if that is possible. So the query that I had, I mean, the data is fantastic, and I was hoping to see something like that, and I'm very happy that you showed exactly what was the plan for the institute. So these mice, these are the mutant mice, they require a specific diet, and you generated a jelly which has amla ki rasayan and ras sindoor. But the animals have to be fed on the same jelly for so long duration, maybe like 6 months or so. So how do you make sure that the nutritional requirement of the animals are taken care by that jelly? And how do you really maintain them eager to eat that food? Because they grew on something else, some sort of pellet diets. Jelly is a new food form for them. And
that's right. So,
yeah, there's one more question. Since the talk was so exciting, so I have been piling up a lot of queries. The second query is that you have a very good data for amla ki rasayan and for ras sindoor. Both of those rasayan we have been following very strongly. Generally, when we see a strong pharmacology data like that, we generally hope to see a dose dependence data. What you've shown today is a single dose data, and I hope that's a human dose equivalent data. But do you have a data for some lower dose or higher dose, and can you titrate a response in a dose-dependent manner? Because that will make things much rosier, crispier, and acceptable people across the world. Thank you.
Yeah, thank you. These are fantastic questions, and thank you first of all for inviting me and given me an opportunity to share our data with you. With regard to your first question on the special diet, there are no special diet. We feed the animal with the same diet, and what we do is that we withdraw these jellies were given in addition to the normal diet. What we do is that we remove the water bottle from the cage and keep the jelly. Therefore, the animal feel thirsty, they eat this jelly, and along with the jelly, these rasayanas also taken. So that's what it is. And once they get used to this chocolate, cocoa, whatever, and then they get addicted, and then every day we keep one jelly, and then one cube of it, and the animal pretty much consume it. And therefore, it is not something that we are injecting. It is a voluntary eating behavior. There may be some wastage, but we assume that 90% of the rasayana that is given is consumed by these animals. That's one limitation of the study. Because as you know pretty well, these rasayanas have to be taken, and these are the metabolites that come out after the digestion are the ones that are expected to function. So it cannot be given in any other means. It has the animal have to eat voluntarily. That's the first part. So there are no dietary restriction, except that we starve them for water, so therefore they are able to feed on the jelly. With regard to your second question on the dosage, yes, I mean, that's an interesting aspect. And as you know that it's a time-consuming work. You have animals fed for about 6 months, and then you have to see the end result. So what we have done, doses, and then see whether it has a much better effect, or very high dose may have any side effect or not. This is something that we need to do. We have not done yet, but certainly that is something that we are planning that we should do. Yeah.
Thank you very much, sir. This is fantastic. I just want to add one more point, because one of the formulation you have used is ras sindoora, which contains heavy metals. And most of the people outside Ayurveda world does not really like to consider heavy metals as a safe option. So since you are working from a neutral Biological Research Institute. It would be very exciting to hear from you. Did you observe any nephrotoxicity? Did you observe any neurotoxicity in the animal who were fed with these compounds for 6-month duration?
Yeah, absolutely. I mean, the outcome is that the animals are better with this formulation. I think it is more to do with how do you make the formulation.
Absolutely.
Perhaps that takes away the worry that it would be toxic. And in the formulation that we have used, that we gotten from the Kottakkal Arya-Vaidyasala, I mean, they are only positive effects. We did not see any negative effect on the animal. In fact, we did this study even with a wild type animal to see whether that could be toxic. In fact, if you feed the animals, the aged animals with this Rasindur, they get better in their cognitive abilities as they age. So certainly it has only positive effect. We have not seen, we have done a lot of readouts. We have looked at Ishak factor, we have looked at oxidative stress marker, we looked at inflammatory markers, we have done cognitive analysis, we have done dendritic spine abnormalities. Nowhere we could see any abnormal effect of this Rasindur in the animals. Yeah, that I can say with confidence.
Sir, thank you for this endorsement, and I'll ask my fellow colleagues who practice Ayurveda here to give a very strong round of applause for sir for saying something about which we have been talking about, saying that all the heavy metal based bhasmas are safe at the same time efficacious. So I appreciate that. We do have one more query for you from the principal of our Ayurvedic college, and he's going to talk something about Sindura.
Oh, sir, are you easy in Hindi? I will speak in Hindi, sir. I will speak in Hindi. Can you understand it?
Fine. Yeah, please. Yeah.
सर रस सिंदूर, ये रस सिंदूर दो इसमें कंपाउंड होते हैं, एक मर्क्यूरस क्लोराइड और एक मर्क्यूरिक क्लोराइड. Mercurous chloride is useful for human body, and mercuric chloride is mercuric sulfide or mercuric sulfide. Mercurous sulfide is how much is there? You have any study that how much because more than 30% mercuric sulfide is harmful. Yes, sir.
Yeah, I certainly we have HPLC data for this, all the components of this. So upfront, I cannot remember and, tell you this, but certainly by, looking at the data, maybe by tomorrow or so, I can share with you. If you can just email me, I can share all the data. So every formulation that we have used, we have done an HPLC analysis to verify what are the components that are there, what level. We'll be able to share the data. I can't recollect immediately. Sorry about it.
थैंक यू सर. एक और क्वेश्चन है कि आपने जो माइस की डोज है वो 41 mg पर kg और ह्यूमन बॉडी में जो उसकी डोज हो गई, ह्यूमन के लिए जो डोज है वो 120 से 250 यानी एक रत्ती से दो रत्ती तक की डोज है. तो ये 41 mg पर kg के हिसाब से कुछ ज़्यादा नहीं? आई थिंक इट इज मोर.
No, we have used which one? ये रससिंदूर है ना आपका क्वेश्चन? रससिंदूर में?
Yes, only रससिंदूर.
हाँ, रससिंदूर we have used about 40 milligram per kg body weight per day. That was the mouse dose.
Sir, you should compare it. I think it is more dose for mice according to weight. आधा क्वेश्चन मेरा अनुराग जी ने कंप्लीट कर दिया. देयर इज नो साइड इफेक्ट. इट इज़ वेरी गुड और इट प्रूव्स द भस्म आर हेल्पफुल एंड यूजफुल फॉर ह्यूमन बॉडी. थैंक यू सर.
एब्सोल्युटली आई अंडरस्टैंड.
मैं सभी छात्रों से एक रिक्वेस्ट करना चाहूँगा कि आप सब इस सेमिनार को बहुत ध्यान से सुनिए. आपके लिए बहुत क्वेश्चन है. अगर आप ध्यान से सुनेंगे तो आपके पास में बहुत सारे क्वेश्चन होने चाहिए और ऐसा मौका बार बार नहीं आता है. आपको इसको यूज करना चाहिए. थैंक यू.
नमस्कार. थैंक यू वेरी मच.
Thank you so much, sir. It was a pleasure having you here with us through virtual mode. We'll take your leave now, sir.
Thank you very much. I look forward to visit to the university as well. Thank you.
May I now invite his address on the topic Ethnopharmacology of Endophytic Fungi. Professor Delfino holds a position of Associate Professor of Pharmaceutical Biology at the Department of Medicine and Surgery, University of Perugia, Italy. His research activity has been focused on the study of generation of natural killer cells from bone marrow progenitors and pharmacological modulation of thymus apoptosis, with particular attention to the physiological and pharmacological role of glucocorticoids in these processes and on ethnopharmacology of arthritis and cancer. It's a pleasure to have you here, sir. Thank you.
Thank you. Thank you for this nice presentation. Ladies and gentlemen, good afternoon. Love, passion, tradition, hospitality, what I've seen here in Patanjali, and I honestly couldn't expect better. So I really would like to thank the organizers for this kind invitation. And it's my third time in India, and for sure, this is my best time in India. As you know, I am from Italy. My city is Perugia, but I'm not going to speak about Italian herbs. I'm going to speak about a Bangladesh fungi. And my story starts with this picture that you can see here, that was taken in the ghost city of Panam in Bangladesh. I was invited in Bangladesh a few years ago together with, for example, Cobus Helof, that you know, because he spoke yesterday. And in that occasion, I and Professor Abdul Mazid from the Faculty of Pharmacy at the University of Dhaka planned a collaboration on endophytic fungi that he was studying. I start with the definition of endophyte. This was given by de Bury in 1866. An endophyte is a fungal or bacterial microorganism that colonize various interior parts of plants, causing no apparent pathogenic effects on its host plants. So endophytic fungi in some way establish a symbiotic relationship with an host plant, and with the help of both sides. The host plants helps endophytic fungi to grow, to be fed, and in return, endophytic fungi protect host plants from abiotic factors such as high temperature, salinity, the harmful effects of lights, or also from biotic factors such as herbivores, nematodes, bacteria, pathogens. This symbiotic relationship is useful for both. Endophytic fungi produce a lot of secondary metabolites that help the plant to defend, to increase tolerance to stress, to improving the immune system function of the plant, to aiding substance absorption, and to bolster defenses against diseases. So it's a very useful relationships. Of course, these secondary metabolites can be exploited also as drugs for many diseases, including cancer. And the story of cancer treated with this kind of substances is long and important. As you can see here, many secondary metabolites were exploited for this purpose, including alkaloids, terpenes, and so on, and many others. And the number of secondary metabolites is increasing over years, as you can see in this picture. There are many examples of natural products that are useful, very useful in the therapy of cancer. Paclitaxel is maybe the most important one. Is used in the treatment of ovarian cancer, breast cancer, non-small cell lung cancer, and Kaposis sarcoma, or the derivatives of camptothecin, topotecan, good for cervical cancer, ovarian cancer, and NSCLC, and irinotecan, good for colorectal cancer. And the vinblastine and vincristine and their derivatives, vinorelbin and vindesins, used for testicular cancer, breast cancer, Kaposis sarcoma, lung cancer, lymphomas, and leukemias. And I am going to talk about leukemias today. And our work started from the symbiotic cooperation between the plant Rauwolfia serpentina and the endophytic fungus Cladosporium. Rauwolfia serpentina is well known in all Asia. Yesterday, a colleague talked about it. And from Rauwolfia serpentina is used in traditional Chinese medicine, and also in India. I guess that Mahatma Gandhi used to an extract of Rauwolfia serpentina as a tranquilizer. Rauwolfia serpentina produces a lot of metabolites. For example, reserpine, which depletes the ganglionic terminals from norepinephrine, and so it's an inhibitor of the sympathetic nervous system, determining, for example, influencing the heart rate, decreasing the blood pressure, and also having a powerful sedative effect. Other compounds are raubasin and yoimbin, which are vasodilator. And in the past, also imalin has been used for treating cardiac arrhythmias. So Professor Mazid grew inside this plant the Cladosporium, which is a common food contaminant. And then he separated many different secondary metabolites, and among these, anhydrofuserubin and fuserubin, which are the metabolites I have studied. These two are two naphthoquinones, differing only for an oxidryl group and a proton group. And when you put these two metabolites together with leukemic cells, I have used a acute myeloid leukemia cell line, which is interesting because it has a mutation in nucleophosmin gene, one of the most common mutations causative of this type of leukemia. So when fuserubin and anhydrofuserubin were cultured together these cells for 24 hours, the number of leukemic cells decreased significantly. Although fusarubin, as you can see on the left, is much more potent. And you can see this also in these time course experiments, in which fusarubin, the red line, after 72 hours, almost completely eliminated all leukemic cells, while anhydrofusarubin, the blue line, was almost ineffective. So there is a decrease in cell number. The decrease in cell number can be the consequence of an inhibition of proliferation or increased apoptosis, increased death, or of both these parameters. So I started working on proliferation by analyzing the cell cycle. And as you can see here, fusarubin was able to significantly decrease the number of cells in the S phase and increase the number of cells in the G2M phase. This means that there is a block in the cell cycle progression. Now, the cell cycle is strongly influenced by two proteins, P53 and P21, that works in concert. They work together. So we have analyzed these two proteins, and what we have seen is that fusarubin, but not anhydrofusarubin, was able to increase the expression of P21 and also to increase the expression of P53 at a certain concentration. Now, P53, P53, P21 is an important pathway which is influenced by another important pathway, which is the MAP kinase pathway. So we analyzed the MAP kinase pathway, and we have seen that that fusarubin was able to induce a significant decrease in the phosphorylation of ERK, which this decrease stabilizes P21, and also fusarubin determined a significant increase in the phosphorylation of P38. Also, the phosphorylation of P38 is a stabilizer for P21 protein. So this is the reason why there was an increase of P21 protein expression. Additionally, fusarubin was able to significantly decrease the phosphorylation of AKT, which is important for the translocation of P21 in the nucleus, so from the nucleus to the cytoplasm. So this decrease accumulated P21 in the nucleus, where it could function as a tumor suppressor gene. So let's go to the first conclusion. Fusarubin determines an increased expression of both P21 and P53. Under these conditions, P21 blocks the cell cycle progression and allows cells to undergo senescence or apoptosis. Let's go to the second parameters. What about apoptosis? As you can see here, fusarubin was able to significantly increase the apoptosis after 24 hours of treatment. Is not a big increase. The apoptosis goes from 2% to 8%. So only 8 cells out of 100 cells undergo to apoptosis. But anyway, this increase is significant. Now, simplifying, the apoptosis pathways that we know are basically two. One is the extrinsic and the other is the intrinsic pathway. In the extrinsic pathway, there is a soluble ligand, such as Fas ligand, that binds to its receptor, in this case, Fas receptor, that recruits the adaptor molecule FAD, that recruit and activate caspase 8, a proteolytic enzyme. In the intrinsic pathway, there is usually an internal damage, for example, a DNA damage, that through the involvement of a P53 protein shift the balance between anti-apoptotic proteins such as BCL-XL and pro-apoptotic proteins such as PUMA in favor of the pro-apoptotic molecules. This brings to the release of cytochrome C from the mitochondria and to the activation of caspase 9, a sister caspase. At this point, the two pathways converge to activate caspase 3 that leads to apoptosis. Now, we have examined the caspase 3 as a confirmation of the apoptotic process, and also active caspase 8 and caspase 9 to see if the process belongs to the extrinsic or the intrinsic pathway. And these are the results. We have activation of caspase 3, so the apoptosis is going on. And this apoptosis depends on activation of caspase 8, but not caspase 9. So it's the extrinsic pathway to be involved, which is quite surprising because P53 is usually linked with the intrinsic apoptotic pathway. To confirm this, we have monitored the expression of Fas ligand by real time, and as you can see, there is a significant increase in Fas ligand expression. And also, Fas ligand, I remind you, that belongs to the extrinsic pathway, and also the expression of BCL-XL and PUMA that belong to the intrinsic pathway that, as you can see, is not modulated by fusarubin or anhydrofusarubin. So, in conclusion, fusarubin on acute myeloid leukemic cells determine an overexpression of P53, of P21, and a block of proliferation. On the other hand, the same molecule determine an increased expression of Fas ligand that activates caspase 8 and bring to an increase of apoptosis. The question is, are these two independent mechanisms, or are they in some way interdependent? And the other question is, maybe there are two cell populations with a different responsiveness to fusarubin. To test this, we made a flow cytometric analysis of cells treated for 72 hours with fusarubin, and effectively we found two different populations. One made of smaller cells, which are apoptotic cells, and the other made of bigger cells, which are not apoptotic. However, the bigger cells, as you can see here, they disappear as time goes by. And so maybe this is a pre-apoptotic population with a different responsiveness compared to the apoptotic population you have seen. So in conclusion, fusarubin blocks cell cycle progression through a p53, p21 dependent pathway and promotes apoptosis through a caspase 8 dependent extrinsic pathway. Fusarubin identifies two apparently identical leukemic cells with an unseen different susceptibility to apoptosis. I like to leave you with a take-home message, which is a question. Do we always know if herbal compounds come from medicinal plant or from their endophytes? So, I mean, sometimes it's difficult to differentiate between secondary metabolites produced by endophytes in nature and metabolites produced by medicinal plant. I like to conclude acknowledging the people that worked on this project. Professor Mazid Dedi's group from University of Dhaka, Isabella Muscari in my lab, Sabrina Doricio that make most of the experiments. Thank you very much for your attention.
Yeah, go ahead.
Thank you very much for your insightful talk, sir. My question is, as we know, most of these secondary metabolites are synthesized by the specific biosynthetic gene clusters, and those biosynthetic gene clusters are tightly regulated for their expression. So, is there any chemical cue that is coming from the host plant that is actually triggering the expression of this biosynthetic gene cluster, or is a constitutive expression? And in regard to this question, is there any other source from where this cladosporium was found, which is also producing this particular secondary metabolite?
Yeah. Well, thank you for the question. If I understood the question, you know, this is a symbiotic relationship. So there is an interchangeable type of influence. For sure, endophytes influence the host plant, but also the host plant influence the endophytes. I don't know which are, in this case, the genes or the metabolites that can influence the endophytic fungi, but these happen for sure because endophytic fungi in different host plants produce different type of secondary metabolites. Regarding the second question, cladosporium is not essential as endophytic fungi to produce fusarubin or anhydrofusarubin. For example, this kind of secondary metabolites can be produced also from other endophytic fungi, such as that of the Fusarium species. And this is another confirmation that this symbiotic relationship depends on fungi, but also on the host plants.
Professor Delfino, thank you very much for a fantastic talk. We really appreciate that you chose to come here and spend some time with us, including the morning yoga. That was really nice.
Well, you know,
let's clap for him again.
It's a gift. It's a gift from you. I mean, this is a gift from you.
Thank you very much indeed for being so humble. The one technical query I had, I mean, I didn't know really before your talk that one could use endophytic fungi to use this sort of application. So my question was does the fungus is specific to a given plant, or you can have that fungus given to multiple plants and could generate a host of different metabolites?
Yeah. Well, I'm not the right person to answer this question, because this part was made by Professor Mazid in Bangladesh. We just got, the metabolites. But what I think is that this is just a system used by Professor Mazid, but it is in some way interchangeable. But if you change, you get different secondary metabolites.
For example,
this is my feeling, although, I don't work on this.
So does the host plant play any role in the amount of secondary metabolite or the type of secondary metabolite made by the fungus?
I think so. But as I said, maybe I'm not the right person to answer this question.
And as usual, my second question is slightly more historical. You gave a small little mention about Mahatma Gandhi and Sarpaganda. So I was wondering that where you learned that Mahatma Gandhi ji was using Sarpaganda as a tranquilizer. So if you have some information,
I must admit that I found this information in Wikipedia.
In Wikipedia?
Yes. So, but I think that with the life Mahatma Gandhi had, probably he really needed a lot of tranquilizer.
Okay. Thank you very much for that understanding. Thank you very much.
Thank you. Thank you.
Thank you so much, sir, for sharing your insightful opinion on the matter. Moving on, the next eminent speaker, which we're going to hear today, is Professor Robert Verpute from Leiden, the Netherlands. Professor Robert Verpute is the Professor Emeritus of Pharmacognosy, University of Leiden. His research activity has been focused on the study of the generation of NK cells from bone marrow progenitors. His research interests are in biosynthesis and metabolic engineering of plant secondary metabolism, metabolomics, medicinal plants, isolation, and identification of biologically active compounds. He is the author and co-author of 810 plus scientific papers, 4 books, and 7 patent applications. Let's give him a warm welcome. Sir, can you hear me?
Okay, you hear me?
Hello, sir.
Can you hear me?
Yes, sir, I can hear you.
Okay. Anyway, now I have to...
Are we audible to you, sir?
Yeah, wait a minute. Share. Where is this one? You see my? Do you see my presentation?
We can see you, sir. Not your presentation. No, we can't see your presentation yet, sir.
You see my slide?
No.
Let me see what to do. Okay. I want to share the screen. But where is that? Zoom.
Try to share your screen, sir.
Good, share screen.
There is an option, yes, sir.
Now we're getting here.
Yes, sir. Now it has started, sir. We can see the screen.
Okay, so now you see the slide, first one? Hello?
Yes, sir.
Do you hear me?
Yes, sir.
Yeah?
We can see your slides, sir. Please carry on.
Okay, good. So, anyway. So, the title, as you see, is Food for Health and Food for Thought. And my aim today is to give some food for thought for how to develop our research in the field of natural products, because I think there's really some things we have to keep in mind in making the right choices, because research is dependent on the money, the grants you have, and that is also the most limiting factor in what we are doing. So by really thinking ahead about what are the possibilities, what are the chances, what are the needs for the world, etc., how to choose the subject of your work. So anyway, it's about plants, of course, because we all have our plant work. And this is something which we share with our ancestors as well. They knew, and like we know now, is that plants can kill you fast, like the cyanide and strychnine. They can also kill you slowly, like opium and tobacco, when you get addicted. They please you by the colors and the scent of the flowers. Plants may warm you when making fire, dress you when making our clothes, feed you, obvious, we have to eat all kinds of things to stay alive, mostly of plant origin. And finally, plants can also cure us from all kinds of diseases. If you look at the plants which through this evolution of humanity has been found, then there's about 30 species which are used for our staple food. About 100 species which are used for the fruits, some 100 species which are used for vegetables, then 50 which are for the isolation of compounds which are used for different purposes that we'll come to later. And then there's some 40,000 to 70,000 which are used medicinally. That's about 20–25% of all the plants have a medicinal use somewhere in this world. And finally, then there's the plants used for fiber, for paper, wood, for spices, ornamental, etc., etc. So there are many, many, many different applications. So major ones are here in terms of the applications. We have medicines, we have cosmetics, we have nutraceuticals, and we have agrochemicals. All four are subject of major industrial activities all over the world. But there's some difference between these. Although they all come from plants, they have different characteristics based on the use. If you look at the value of the end product, medicine have a very high value. Why? Because it's quite a lot of money involved in to produce and then also to find novel compounds from nature for medicinal uses. The activity is usually in the nanomolar range, that means a very, very active compounds. And the total amount produced worldwide is maybe only in kilogram to ton-wise amounts. If you look to cosmetics, high price, but there we're talking about something which is not so very active. Why? Yeah, because if it's too active, it could be a problem of getting intoxications too easily. So we need to have compounds which are medium strength in terms of biological activity. And the amounts, again, is in the same, like for the medicines, it's in kilogram to tons production. So it's not big agriculture, it's more horticulture productions. If you go to the food, we're talking to something different, because now we're talking about things which we have to eat every day, or we add it some time per day. And the value cannot be very high, because our food is already expensive for most people, and to have things which are more expensive than the food, because of presence of certain compounds, will not really fit into use into the food area. The activity, again, is also here, it's millimolar amounts. And then you can see the difference with the previous one is that it's tons to bulk production. So there's some examples of plants which are really used in huge amounts. And then the last one is the agrochemicals, the compounds we use to protect our plants from infections, from insects, from herbivores, etc. There, the farmer has very little money to spend on the fields he has under his jurisdiction. they need something which is against the pests and diseases, which is very, very active, and then very cheap, and the production is really ton-wise. So you see, each of them have differences in the economics. And that is not only reflected in the final product, but also means the amount of activities in research is mostly on the medicines and the cosmetics. The food companies, and I come to that later, are now also interested because they want to have extra value, and agrochemicals. If you really make the calculations, you can see that it will be very difficult to have on the major staple crops the use of any natural products as an agrochemical to protect the crops. But maybe for more precise horticulture and the bio grown vegetable, that there is some market there. If you take the traditional medicine and modern science, then there's always a discussion, as we all of us know very well as pharmacologists and natural products people, that the chemists and the pharmacologists, they want to have a single compound. So they go for trying to find the compound which is active in traditional medicine, for example. But we have to keep in mind that many of the traditional medicines are even mixtures of different plants, so that it's not really one single compound in such traditional medicine which is the compound that complains the total activity of such a plant. Of course, there are examples where that is the case, like the artemisinin. But even there, it is known that the traditional medicine seems to be more active than the pure compound, and we come to that later. Synergy is a very important point on this. If you think about the medicinal plants, then it's more a holistic approach by the medical doctor or by the traditional healer. And the way of trying to find one single compound which is contain everything is not really the best way to do it, in my view. And again, we'll go into the depths later on. So the holistic approach means that the most logical thing to study traditional medicine is to start with clinical trials to see if you can see that the person with a certain disease is cured, or at least has a better situation than without the drug. And that's where systems biology comes to in the picture, and I will briefly come back to that as well later. So that food has an effect on our health is obvious, and the vitamins, antioxidants, prevention of cardiovascular diseases, cholesterol lowering, cancer preventing, GI tract health, all these sort of things are known to be affected by various medicinal plants and even by things we just eat as food. On the other hand, we have also to realize, and that's what many people forget, that everything could be bio, but nature doesn't make all these secondary metabolites to be nice. It's more that they make these compounds to defend themselves against herbivores like we are. And as a result, many of the compounds you can find in plants are carcinogenic. They have liver or kidney toxicity. And some of these compounds, and that's of course is the more advanced, the use of Western medicine in combination with the traditional medicine, there is a risk that there is compounds that interfere with each other just by making it faster metabolized, slower metabolized, all these sort of things may happen. And that is where the old knowledge of our ancestors meet with the modern medicine, where there would be some things to keep in mind that are riskful. If you look at the food industry, they have a lot of interest right now in having health-promoting compounds from plants. Why? Because they want to prove that their products are healthy, or healthier than the other company who has maybe not the same variety of a plant which is as rich in certain desirable compounds as the other. So if there's really identified some compounds which are important, then they can also do something on the plant breeding side. Or they can produce the compounds and add these compounds to existing food. In case of compounds which are not good for our health, like the most common example is probably pyrrolizidine alkaloids, they are highly toxic in the long term. And if you want to use this plant type, you need to be sure that these compounds are not present. Anyway, that's a major reason why there's a lot of interest from the food companies nowadays in the traditional medicines and in looking at the health effects of what we eat every day. So being a natural product person, you always wonder, of course, how many compounds are there around us? And there's several people who made estimations about the chemical space. And these estimations run for like from this author, Merger, that there are 10 to the 40s to 10 to the 100s small molecules possible. So by just taking the major compounds, carbon, hydrogen, oxygen, nitrogen, and then maybe sulfate, sulfur, and then keep a molecular weight of about 1,000, there's enormous amounts of an infinite amount of small molecules possible. But of this full estimation of the chemical space, there's only very few drugs, because maybe there are 1,000 compounds which are used as medicine, which are coming from nature. And then they calculated in this paper that the chance that you find an active compound is 1 in 10 to the 14th. When you measure the effect on all targets available and all in parallel. That means there's enormous small chance. So if you have money, you probably better put it into the state lottery than in going into the chance of 1 in 10 to the 14th to find a new drug. If we look at the chemical space which is known now, then here's 3 databases which are of interest to look at. This is Medlin, which have about 850,000 compounds. It's PubChem, which has about 93 million compounds, and that's not natural products, of course, it's mainly synthetic products. And then if you really want to know the natural products, particularly sugar metabolites or specialized metabolites, whatever you want to call them, there's about 328,000 plus, and that's increasing every year, of course. But you see, these numbers are only very, very, very small if compared to the total chemical space available. So if we want to do bioprospecting, which is the systemic search for organisms, for genes, for other compounds, for designs that might have potential use, there is an enormous amount of possibility around us that we need to find a way to focus to be successful in getting a novel drug or whatever is your goal. So another way to look at this is how many compounds would there be in nature, because these 93 million, which are mostly synthetic, we do not care. But how many compounds are there around us? And one estimation we can make is when we see how many different species of organisms there are in the world. And even that is, of course, part an estimation, but at least most of them are based on certain collection which are having these different plants or microbes, etc., and identify them as individual species. Then we see there's about 250,000 species of plants, and some say it's 300,000, but it doesn't really matter. It's the order of magnitude which is important. The lower plants are a lower number. It's only 12,000. The vertebrates is also about 12,000 different species. But here's an interesting one, 32 million different species. Algae comes to 10 million, fungi, yeah, that's difficult to, because with all the new sources for isolation of microorganisms from the endophytes and whatever you have, and the GI tract biome. But anyway, 1.5 million, and for prokaryotes, 1.5 million as well, but it could be tenfold more, it could be 100-fold more. There's a lot of things there which are underestimated probably in the lower part of this slide. But altogether, it means that there's 10 to 100 million different species. Now, if you assume that every organism can make one unique compound, there must be 10 to 100 million natural products. That's much, much, much more than the ones I mentioned before, which is 328,000. There's many, many times more. In other words, there's still a lot of things to be discovered in nature. And at least I think nowadays the estimation is that there's about 5,000 new compounds found every year, but still you can see that it will be hundreds of years before we really have all the compounds which are around us. So still much to discover. So if you look at medicines, if compared to food, we already mentioned the differences in terms of the economy, but there's more, because in medicine, Western medical science want to have a single pure compound working on a well-defined target. The toxicology is addressed in the registration, and then there should be evidence for activity to be able to make any claims. So we have to go to the FDA or EMA to get your files evaluated, and they make the conclusion that, okay, this seems to be an interesting mixture of compounds in the case of a plant, in food, or when it's a genetic compound, that the single compound is safe and does what it promises. Problem here is this very high development cost to come to a single molecule drug application. Also, we have to realize that in Dutch language, the word for medicine is geneesmiddel. That's something that cures you. But only very few medicines cure. Most medicines, they just are symptomatic treatment of high blood pressure or high cholesterol or whatever, but not really curing. They're making conditions such that the risk of getting a disease are smaller, so it's part of prevention as well. If you look at the food and the plants for health, then we're talking about complex mixtures, which are highly variable. It's the synergy, the product which is happening. There's the quality control, which you have to fit the activity as well. The development costs are also very high because you still have to do a lot of things, but probably not as high as you're coming to a pure compound. And it cannot be too high because if it is food, the price must be reasonable. And you cannot ask for a cauliflower which costs 100 euros. It's like you can get a pill which maybe costs 100 euros, where in that case, if it's a pure compound, will be accepted. The insurance company will pay for it. If it will be food, no one will be think about it to pay for that. But here also is very important prevention. And in making the choices on what we're doing, we have to think about this high development cost, and we have to think about low cost food, how to deal with that. Because here there's a direct effect for the people who eat it every day, also for the poor people. To the left, with the medical things, it's for the rich people who have the money to buy all kinds of medicines to camouflage their bad living habits. a bad lifestyle. You all know these statistics of a new one in crack every year, more or less, become new one. And what the major message here is that about half of all new drugs which are coming out from the market in this period are natural products or natural product derived or synthetic analogues of the natural product. Whereas the pure chemical ones, where there's no relationship other than that the target could be also working for a natural product, but the structures are completely from either the calculations of what is the active site and what is the best binding to that active site, etc., and then make all these synthetic drugs. But that's not the most important part. It's the good news for us as natural products people is that about half of all novel drugs are natural product derived. But the bad news for all of us, the number of novel drugs is decreasing dramatically. And we come to this in later on as well. But just think about the pandemic disease we had around the drugs companies producing medicine. They went the way of biologicals because you can make apparently a new vaccine for a new disease within a year, but to think about a new compound, to develop that will take much longer. But anyway, just showing here, over the years, the percentage of the natural products of the novel drugs developed during these years, from 1981 to 2009, you can see it is somewhere around the 40%, going up and down. Some years as many, other years are bad, and over the long term at least, it is some sort of circular things in there that work. Anyway, the drug companies work according to this principle, what I said before. It's the paradigm of modern drug development. Single compound, single target. But there's also a negative side. This is from an Iyer, research director in GSK, who said some 10 years ago that the vast majority of drugs, that's more than 90%, only work in 30 or to 50% of the people. In other words, if you want to say this in a negative way, that at least half of the people taking medicine have no other effect than maybe a side effect, but they're not really doing anything for their health. That is. And of course, there's now a lot of discussion about the DNA profiles, which could be used to make this percentage that have a benefit will go up. But the problem is, would the pharmaceutical companies be interested to cut this? Because if you cut it to the 30% which has a profit, which have a benefit from the medicine, it means that you lose 70% of your market. So you can see the work on the personalized medicine based on DNA profiles is mainly in the academic spheres, in institutes which have no profit. For the pharmaceutical company, it's even better if you can show that if it's even works in 10%, that gain is so high that it's still worthwhile to let 90% of the people taking something they don't really need. But there's more to this, and that is from this. Also, Leonidas, you probably know him. He has written many, many papers. He is a medical doctor and mathematician. And he showed in this particular paper, which was the title, Why Most Published Research Findings Are False. And he shows by taking a number of papers out of the literature from top journals like Science and Nature, and then show that there are so many variables that you cannot draw the conclusions which were drawn in these papers. So we say for many current scientific research fields, claimed research findings may often be simple, accurate measures of the prevailing bias. And two, simulations show that for most study designs and settings, it's more likely for a research claim to be false than true. And he estimates roughly about 80% of all the available papers in top journals are not necessarily true. And not by purpose, but just by the fact that there's too many variables that are not taken into account. And you all know now the discussion that in clinical trials, for example, women are not included to the same extent as men. 80% is male, 20% is female. Why? Because women have a variable biochemistry over the months. You know, all the problems of having a need for much more female patients, do it with the men and then say it will be okay for women. But that's one example where you can see that all these papers which are published are sometimes so skewed that you cannot draw the conclusions they say. Anyway, those guys in Amazonia had 50,000 different plant species around them. And they knew two species, the strychnine species and another Venus palmatius species that could be used to make an arrow poison called curare. And the ingenious thing about this is that these arrows, which were dipped into the arrow poison, is deadly poisonous by injection. For a human being, it's only a few milligrams needed, depending on which compound you take, to kill you. But you can eat a kilogram of curare and nothing happens. Why? Because the compounds which are active are binary, quaternary alkaloids, which are unable to be taken up in the body. Bioavailability is zero. And in case of using it as an arrow poison, it goes directly into the blood, and whatever animal is getting this arrow in his body, it will die on the spot. How did the people find this? They had no HPLC, they had no NMR, they had no nothing. Yeah, okay, you can say it's pure guessing. True, could be. But why then is that also in Central Africa, there is a strychnine species used by the local people as an arrow poison containing these type of alkaloids. And again, go further east to Malaysia, also there was curare-like agents made out of strychnine. So 3 times independently, people found this principle of make an arrow poison, and it is good for hunting, and it's good and not bad for your health. So, seeing this, you get the feeling those people are very clever. They have been able to get these things found, and how? We do not know. You don't eat 50,000 plants, leaves, or whatever species. But they did it. They have some way to find that these plants could be used for this purpose. If you look at the development then of science, in the great past, they found these plants. In the 19th century, people started to look at the pharmacology in animals. And then in the 20th century, they came to organs and to cells. And then at the end of the last century and in the new century, we're looking at the level of the molecules. We go from super holistic, where people have the whole forest around them and to select from, to the super reductionist approach, where we try to cut as fast as possible to one single compound. Anyway, the curare is a beautiful example of how the science has developed, because in the 1940s they did a lot of the chemistry, they came to this point that it is two quaternion nitrogens on a certain distance which are needed, and then synthetic drugs were made in the 1950s, which could be used instead of the very complex structures coming from the nature. But that's the whole pharmacy development you can find back in the Curara. Anyway, if you look at the whole process of drug development, you start always the finding of the lead. And when you have a lead, then you can still look at minor changes in the structure if that could help to make the compound more active. And then finally, you have to go to the clinical trial. This part is completely secret. It's happening in pharmaceutical companies, but they want to save this because from this moment, you have to go to a public announcement of what you're going to do in the clinical trials. So this one you can do in secret. Here you need to come with the arguments why you can do the clinical trials. But here you must take your patent, and 1 year of patent too early means that you lose the patent protection for 1 year, and then you can make talk about billions of euros in losses, in fact. Anyway, then finally, when it goes through all the clinical phases, you have the new drug application, and that goes to the FDA, EMA, and whatever organizations you have all over the world, to the national or international level, do the registration of medicines. Originally, say about 10 years ago, 20 years ago maybe, there was 100,000 compounds needed for one single drug, new drug. Now this could be even higher. This is probably up to 1 million. Why? Because most diseases where there is any reason to find a drug has been addressed, and there are drugs which are doing the job, maybe not as good as you want, maybe better one, but still to have the total cost of making the development, it is inhibiting here the drug discovery. For the 21 novel drugs like in 2003, and you saw before this graph, 40% of the drugs must have millions of compounds tested before you find a novel drug. You can also see the time scale. This is 1 to 2 years to get it from the hit to lead, then 1 or 2 years more to really get the final composition, and then 1 to 3 years to see which one is the clinical candidate and doing the toxicity so you can enter this next step, etc. But you see, there's a lot of time here, and the whole process from here to here is between 10 and 15 years. And the cost, you can see here, in the gray ones is the 1970s, 1980s, and the black one is in the, say, the 2000, 2010, that's the drugs which has come out in the past 10, 15 years, more or less. And for the pre-human phase, you need already 1 billion euros. In clinical trials, even more, 1.5. So you see this number, 2.5 something, in cost. Now, think about how much is your budget at the university. It's so low that this whole thing can never be done in our labs. At best, we can score in the pre-human phase that we have some hits, and what the hit is worse, yeah, that depends how activity there is involved, and then what dose, etc. But selling your hits to the pharmaceutical industry gives you some thousand euros, but nothing more, because then they have the money to really start to look at it. So you can also see here the problem, like with the pandemic, as an editor of journals, you get a lot of papers who want to make a review and say what has been done already. I had hundreds probably have been sent into different journals and have been published. And all want to do the drug development to interfere with the COVID. But you can see with this development time of 10 to 15 years, in 10 to 15 years, then no one knows anymore what COVID was because it has been introduced then as a common disease. And that's why the lesson is as well, when there was SARS, there was a lot of work done by a lot of people, ending up with a number of compounds which have some activity which would be to treat SARS. But then SARS was not really so effective as the COVID was, and then it stopped, and all these compounds are there, and no one has ever finished it. We lost 10 years because of not having the guts to continue supporting the research to come at least further in this picture from pre-human to the clinical one. So when the COVID came, we were empty-handed with a lot of ideas, but none of them really good enough to be used. And to really make it to there, it's 10 to 15 years of work. Knowing that the vaccines can be done in 1 year, it's obvious that the vaccination is beating the natural products here completely. Of course, vaccines are also natural product, but it's a different thing from a small molecule and a large molecule as used in vaccines. Anyway, so we are in a session where ethnopharmacology is important, and all these sort of things has to do with ethnopharmacology, of course. And if you want to study traditional medicine, you can have two different goals, or you can have them both at the same time, of course. You can try to find a novel drug based on the use of traditional medicine, or we go for this one, just make clear and prove that the drug traditionally used has the activity people use it for. If we're able to do that, then you really have, in short term, something to confirm that people are using it already. So fine, and you can say, look, if you want to do it effectively, do it this and this and this. Or in case that it doesn't work, or even worse, that it's toxic and doesn't work, then you can say, please don't use it because it's not good for your health. So this one, I think, is the most important one we should look at. At the same time, as a derivative from here, you can see if there's any active compound which could be used and such. But anyway, what we need to know is the mode of action. We need to know toxicity. We need to have market for activity because we have to have quality assurance. And of course, the toxicity is the most important one. So we really should, in our work, choose methods that at an early stage can say, okay, this is highly toxic, not only acute, but particularly long-term toxicity is killing many of the good ideas. So if you first put a lot of effort in showing that it is a beautiful thing which does what you want, and the next is to prove the toxicity, and it there falls, you have done all this work for nothing. So the toxicity is really, really the entrance to both these areas. In terms of the novel drugs, the problem, of course, is that there's the synergy in products. That means there's no single compound. And this is very likely, in fact, because we know that in traditional medicine, there's certain rules of how to prepare the extract, and often as a mixture of different compounds, which in the Chinese have the different things, which is the major therapeutic one, and there's another one which is a transporter inhibitor or transport and increasing, etc. So that's an idea behind this already. The toxicity has also the point of the variability. It's all kinds of microorganisms on plants are there, important for the quality or not. And contaminations, of course, could be coming from other sources.
Sir, sorry to interrupt you, sir.
Yeah.
I'm afraid we are running short of time, as our slot has already extended by half an hour.
Okay, now you sent me an email saying at 40 minutes, so that's why it's a little bit later now. Okay, let's see.
Yes, sir. Please conclude it faster.
Yeah, okay, now I'll make it shorter. But I had it for 20 minutes and then I get a message to make it 40 minutes. Okay, but
Yeah, it is 20 minutes, sir.
Okay. Anyway, synergy. Two or more agents working together to produce a result not obtainable by any of the agents independently. That's the definition. Basically, 1 + 1 is more than 2. That is the basis of life. So how to prove it now? If you do fractionation, then you lose activity. Isobolograms, this is when you take two compounds together and see if there's activity. Yeah, but which compounds to test? No, there's only one way to do this, is using system biology, correlating compounds with activity. That means activity and metabolomics. So there is a paradigm shift in science, and that is going from the hypothesis in which we had the idea we can explain and make everything, that we gradually start to realize that this does not work always. Like in cancer, there's some doesn't really help always. Plant resistance is much more complex than that, and genomics also is not so easy as people thought originally. Anyway, by combining all this, then you have the system biology. In the system biology, you're an observer. You're not having any hypothesis. You just measure and measure and measure with all possible ways you have. And that's what's called system biology. And the key technology there is metabolomics, the chemical characterization of a phenotype. And the aim of metabolomics is to identify and quantify all metabolites in an organism. Why? Because life is chemistry at work. If you can follow the chemistry of life, you find the ways where you can cure, for example. We like the NMR very much because it's highly reproducible, it's a small amount, it's fast, and its absolute quantitation is very easy. But I'll not go into any details here. The system biology should help us to understand the synergism of products, which compounds are needed to have the activity. And if you start to separate them, you lose, you can then see which compounds are involved. This way we can find novel modes of action. We can confirm the traditional use with an holistic approach. Still, the identification of the active compounds could be difficult because it could be a prodrug, could be a very complex compound. And again, it's against the paradigm of drug discovery, the simple, single target, single compound theory. Give you one example. This is here, it's a microorganism where we found that if you induce biosynthesis of new antibiotics in the microorganism, we get these new peaks. We have high activity because of this induction, and the point was that this compound is the antibiotic is very, very small amounts, very, very, very active. So you don't see it in NMR, but if you look in NMR, this compound is present in huge amounts. Now, why is that? It is synergism. You can hear if you test this antibiotic with the antinelic acid, you see that you get them for many antibiotics, you get a much higher activity. Similar thing for the isoflavonoids. We've shown that these isoflavonoids, they do really inhibit the NOR-A effect pump and thus increase the effect of an antibiotic 4 to 8 folds. Okay, synergism, and this is the crux, produce a result. What is the result? The crux is a longer life, or a lower blood pressure, or killing a microorganism, or whatever. So, in traditional medicine and Western medicine, all have the same goal, that is homeostasis, to get that back. What is that? Homeostasis is the age, the gender, the genetics, the work, the food, the environment. All these things are different for everyone. And if you look for the Western thing, the Western medicine, it's single target, single compound for high cholesterol. So, but no one knows why cholesterol is high and what does that mean for the disease. But if you have the correlation that lower cholesterol, less cardiovascular diseases, then you can give it to half of the world population. And the same here.
Excuse me, sir. I am very
So there's a
sorry.
big difference.
I think we have to cut short the program, the presentation,
Yeah,
because
5
we have
minutes
run
more,
out of time.
but I'm almost finished now.
All right, sir.
So what we need is the synergism and the products in in vivo assays. Clinical trials, closest reality. Animal experiments, same fish, and the C. elegans, Artemisia salina. You see, from very expensive to very simple method which can be used in any laboratory. That's what we need for our work. The message is that to develop novel medicines from the medicinal plants is 10 to 15 years of work. Cost around 2.5 billion euros. So it's better to focus on the used traditional medicine and prove safety and activity and have the things used in the right way. Also, we have personalized medicine, which is normal in many places, but not in the Western medical world. So maybe we can find in this way simple methods. So use system biology approaches on omics tools to study the effect of medicinal plants. That is what I wanted to say. And collaboration makes the impossible possible, just like in synergy. Thank you for your attention. I'm sorry that it was later than so, but
Thank you very much, sir. It was a very enlightening talk. Thank you.
His talk upon protective effect of natural compounds in models of neuroinflammation. Dr. Singh is presently the Associate Professor and Head Department of Pharmacology and Toxicology at National Institute of Pharmaceutical Education and Research, Raebareli. It's a pleasure to have you here, sir. In past, Dr. Singh was associated as a senior research scientist at New Drug Discovery and Research Department at Pharmacology Ranbaxy Research Laboratories. He also served as the Associate Professor of Pharmacology at Amity Institute of Pharmacy, Amity University, Gurgaon.
Hello. So respectful greetings to Shraddha Swamiji and Acharya ji. And thanks a lot to Dr. Varsne for inviting me here and to interact with the faculties of Patanjali, the students of Patanjali, and the scientists across different organizations across Patanjali. Now, let me tell you, this lecture is unique to me because the audiences are a mix of industry people, students, and the faculties together. So this creates a different kinds of environment because wherever we go to conference, that's either academic setup or it's an industry setup. Here it's a mix of all these three together. And that's an opportunity probably for the students, and that you should understand it. You should use this opportunity to explore your thoughts further. Now in today's lecture, I will talk about protective effect of natural compounds in model of neuroinflammation. Now neuroinflammation is an area which even I am working since last 3 years. Prior to that, I had been working on autoimmune, chronic autoimmune disorders and respiratory disorders. So whatever I am doing since last 2 to 3 years, those are the things I am going to share in this seminar. Now, as I said, inflammation is, in general, is present everywhere. Either it's cancer or it's inflammation of gut, central nervous system, respiration, lung inflammation, inflammation in bone, across skin. So there are different kinds of inflammation in different organs. And neuroinflammation is the subject of discussion today. Now, what happens actually in neuroinflammation particularly is it's basically a host immune reaction. We saw in yesterday's presentation there are different kinds of cells in our blood that get stimulated and produce acute inflammation. Now, when this inflammation heals on its own, we say it's a progressive healing, and that body takes care of it. But when it is not healed properly and there is a chronic stimulation of these cells, we say it's a progressive destruction, progressive inflammation. And that causes chronic disorders like all these disorders. So what we will discuss is we will see how we can look at the models of neuroinflammation with respect to in vitro and in vivo, and how we can evaluate the pharmacological effect of some of the natural compounds, which I have done with one example. Now, we had been talking about forward translation, reverse translation. Now, in what we do in forward translation, all of us know. But we look at the disease pathology, and then this knowledge we use to create different models, the cell-based models and the in vivo models, and then this data is translated to clinics. We test those molecules in human. But there are, as you know, like we had been talking about reverse translation. So what we do in reverse translation is we look at this data first, and then come back and establish the disease pathology in vitro or in vivo to give scientific backup to compounds like natural compounds. So this is what we have done here as well. And what we have done is we first, in case of neuroinflammation, first of all, in Alzheimer's disease particularly, we looked at what is the data in healthy versus disease. And what we have seen is these are some of the biomarkers that are being increased in case of neuroinflammation, particularly in Alzheimer's disease, like IL-17, IL-6, TGF-beta, and IL-1 alpha, apart from TNF alpha and others. So these are the important cytokines, like IL-17 is important cytokine in Alzheimer's and across other neurodegenerative disease too. So maybe IL-17 signaling and IL-17 receptors are important in neurodegenerative diseases, and along with all these core cytokines. Now, with this knowledge that these are the biomarkers, endpoint biomarkers, which we need to look at in cell-based assay or in vivo assay, we came back to lab and looked at the literature, and we know from the literature knowledge that tau and amyloid beta pathology, these are the two different hallmarks of Alzheimer's disease. And amyloid beta basically gets deposited into the neurons. And these neurons, they help in transmission of different signals from brain to different parts of the body. So when there is a deposition of amyloid beta, it disturbs synaptic dysfunction. The astrocytic cell population, they are responsible for like vascular supply to different neurons. And whenever there is such deposition, they get damaged and the vascular dysfunction starts. The blood supply to neuron is altered. And the microglia are the resident immune cells of the brain, and they take care of any foreign thing that goes into brain. So this gets stimulated and try to take it out. And in that process, it gets activated and produces inflammation. So this releases inflammatory cytokines and causes oxidative stress. So there are two things. One is neurodegeneration and another one is neuroinflammation. And once both the things are present here, they make a cyclic process and amplify together. So this we have reviewed and published in different papers with what are the targets in these cells, what are the markers in these cells, and what are the targets that we can explore. Now, in vitro models, we have created two different models, the amyloid beta-induced model and metal exposure. Now, sometime back, we had been discussing about metals, so I would like to tell here that metals are an essential element for our brain functions. But there is a dose above which it is toxic. So that LD50 of different metals we need to know, particularly for heavy metals and all. Above that dose, we don't need to go. So we treat with that dose of metal, which is towards higher side, and then look at different models. Why? Because we have seen like A beta and metal exposure, these are the two major causes of A beta deposition, which is reported. And when there is an A beta deposition, it activates ROS, amyloid beta seed formation, tau hyperphosphorylation, and of course inflammation is there. And all this together causes the neuronal death or neuronal damage. So here we wanted to see like what are the biomarkers that are being upregulated, particularly in microglial cell lines, like after activation with A beta. The metal exposed models we have already published. We are working on these models now to validate it and to test certain compounds, natural compounds. I will show one example in this model. So what we have seen is that phospho-P38, phospho-MK2 and phospho-NF-kappa B, it is increased in a time-dependent manner in a 9 microglial cell line on treatment with amyloid beta. And it also causes increase in TNF-alpha and oxidative stress in terms of nitrite. As we see here, there is an activation of IL-17 which we are talking about. There is an activation of IL-17 in A-beta treated animal as compared to control. And to confirm that this response is not non-specific, we have also used other inducers in cell line, like for example, LPS and IFN. So what it shows is in addition to A beta, LPS, and also it shows the activation of IL-17 in N9 microglial cell line. Now this is IL-17 and we have also measured TNF-alpha. So these are the two major cytokines that we see is released by microglial cells. Now when we talk about in vitro models of A beta, there are well reported models for A beta in vitro. But when we talk about in vivo, there are not much confidently worked models. Like there are these two models which are reported in literature. One is transgenic model, the other one is a stereotactic model. The transgenic model, as we know, like yesterday we discussed about the genes and all, there are certain genes which are being upregulated in some disease. So in Alzheimer's also there are certain genes which are being upregulated, and we overexpress those genes in animals and get the disease in these models. So those models are called as transgenic models, and since these are overexpressed models, so they are expensive and it takes time to overexpress those genes in animals. So this is, there are limitations with this model that will increase the cost of drug discovery as well. The stereotactic models, they are invasive and have high mortality because what we do here is we inject A beta into brain. We cut the brain and inject A beta directly into brain. So they have a high mortality rate. So it's like we are testing brain functions and the model itself is like the brain is damaged. So it takes time to revive back. So they are invasive and they have high mortality rates. So we are working on another model which is transnasal model, and this kind of model is reported scarcely in literature, not much. There are only a few reports. So the advantages of these models over these are they are simple, they are non-invasive, and they bypass blood-brain barrier. And transnasal means we insert A beta into the nostrils of animals directly for 4 to 7 days. And we confirm A beta deposition. Now what we have seen is as compared to control animals, there is A beta deposition from 4th day onwards. And this we have confirmed from 4th to 7th day in all the animals individually by histology, by Congo red staining. So when we do not stain with anything else, we only stain with Congo red without any counter stain, then only we can see this amyloid beta deposition. Prior to this, we confirmed with Evans blue transfer across the brain by giving transnasally. Now, this transnasal transfer of any protein or any dye, it does not happen as such. We have to include some of the osmotic agents like mannitol. That will increase the chances of blood-brain permeability and facilitate the transfer of these molecules across blood-brain barrier. So this model we have developed and then went on and tested with the neurobehavioral parameters, because what we are talking is neuroinflammation, but there are neurodegeneration as well once the inflammation amplify. And that leads to different changes in neurobehavioral parameters. So this is one of the tests which is called Morris-Watermass test. And what we do here is it's a tub filled with water. And if you can see here there is a platform. The platform is hidden over the surface of water. We put the animal here, exactly opposite to the platform in another quadrant, and then allow animal to swim. Now, rodents are natural swimmers, and they will try to reach the platform once we train them for some days. Now, they will memorize, once we put the animals here, they will train them and they will memorize that how they reach to platform for certain days. And this kind of memory we say as spatial memory, the learning memory, right? We have different kinds of memory. One is learning memory, and which is in the different parts of the brain. And one is cognition memory, the power to recognize. So this is learning memory. He will learn and memorize it, how to reach the platform. So as we see here in control, all the animals reach the platform, whereas in A-beta treated, dose dependently, there is a decrease in the number of entries to platform zone. Similarly, there is another neurobehavioral test, which is novel object recognition test, also talked by another speaker previously, Professor Ganeshan. And what we do here is we first train the animals with two similar objects. And after training, we change one of the object. And it is the tendency of rodents to explore more time with novel object. So we denote the time explored with novel object, but prior to this, this training has to be done so that the animal is acquainted with these, both these objects together, and he's able to recognize the novel object. So as we see here, as compared to control animals, there is a decrease in this recognition memory of the animals, those with A-beta administration. And after doing the neurobehavioral test, we have isolated the brain, and then we have looked at different parameters, like, for example, the oxidative stress parameters, reactive oxygen species, NO, MDA, and antioxidant catalase protein, and the level of IL-17 as well as IL-6 in the brain homogenate. And what we find is there is an increase, dose-dependent increase in the levels of these parameters in A-beta treated animals, whereas the antioxidant protein, there is a dose-dependent decrease. Now, in this model, once we saw these results, then we tested this quercetin in this model. And quercetin is, why we choose quercetin is, it is known to have different pharmacological activity, like anti-inflammatory, antioxidant, and anti-apoptotic property, anti-cancer properties, and is used as immunobooster. But it has not been tested in this model of neurodegeneration and neuroinflammation. And quercetin, as we see here, it is available across a wide range of food items. So we tested quercetin in this model to see how it behaves in A-beta model. And what we saw is we did the same test after treatment with quercetin. And as we can see, the A-beta treated animals, this is A-beta group. The A-beta treated animals, they do not, the time is not decreased during training days. The time is kept constant. Whereas control group, they learned how to reach the platform. So there is a decrease in time over days. And the quercetin treatment in A-beta exposed model, it significantly reduces time, which is similar to control. So these are all the parameters. These 3 parameters are related to Morris water test, which shows that quercetin is effective, and the novel object recognition test, which also shows that quercetin treatment to animals increase the novel object recognition time. In the same model, we looked at different parameters, inflammatory parameters, TNF-alpha, IL-6, and IL-1 beta in different regions of brain tissue, hippocampus and cortex, and we find a dose-dependent decrease with the treatment of quercetin in all these evaluations for cytokines. With respect to ROS, nitrite, and GSH, which is antioxidant protein, we again find beneficial effects with respect to quercetin, either in hippocampus region or in cortex region. We further found out, like as I said, there are different structural markers of Alzheimer's, like APP, phospho-tau, BACE1, GFAP protein, which is the astrocytic marker, and NRF2, which is antioxidant protein found in these microglial cells. And what we see is treatment with quercetin at 30 and 100 mg per kg to animals. It altered the level of proteins like APP, phospho-tau, BACE1, and GFAP towards beneficial effect in A-beta model. The convoluted staining, which was again done to see whether the A-beta deposition, which we see in A-beta treated animals, is being, has any effect by quercetin. And interestingly, quercetin shows, treatment shows that A-beta, there is an elimination of A-beta from brain tissue. There is a reduced amount of A-beta protein which is present in these animals, in their brain tissue. We also did Nissel's staining to find out the neuronal health, which is a marker of, basically this Nissel's stain. By this stain, we stain the nucleus of neurons, and that shows the density of neuronal population in brain, in particular region. Now in this, you can see the whole brain. Now here, this region of brain is called CA1, CA2, CA3, and this is called DZ region. Particularly in Alzheimer's disease, the CA3 and DZ region is affected, and that there basically the neuronal degeneration happens in these regions of brain. So what we see in CA3 region as compared to control, you can clearly see there is a decrease in neuronal population, which is being restored by quercetin treatment at 30 and 100 mg per kg. And the same is in DZ region. As we can see here, there is a compactness of neurons in control, which is thin in case of A-beta, and it is reversed back by treatment of quercetin. So if we look at the summary of results, we see decreased APP protein, decreased A-beta deposition, improved redox balance, decreased inflammatory markers, decreased tau levels, and all this together causes decreased neuronal death, as we see in histological experiments. So this is what are the preliminary results. Now what we discussed is quercetin is a pure compound, and when you separate it from the extract, the absorbability is problem. It is not absorbed properly because it's a plant product. So we are working on it to how to give quercetin in a proper formulation so that it is absorbed properly, because the bioavailability study shows that only 1% of what is being given is absorbed. So those studies are being done at our end. I would like to acknowledge my institute, the ministry under which my institute works, the Department of Pharmaceuticals, Ministry of Chemicals and Fertilizers, Government of India, and the major workforce in my lab, the MS Pharm and PhD scholars. So out of these, Vaibhav, Junaid, and Avtar, they are working on all these models, A-beta models and testing of quercetin. So thanks a lot to them. Thank you.
Thank you very much, sir. Questions from the audience?
So if you have any questions.
So I have one question from you. So, so bioavailability of quercetin was one question which you already addressed. My other query is about the blood-brain barrier. So about the blood-brain barrier. So are there any studies that have been performed to test quercetin, how effective it is in crossing the blood-brain barrier and then show its effect under the clinical conditions?
So what we have to do is and do the bioavailability study same way as we are doing in blood.
Okay.
To find out what amount goes to brain after administration.
Okay. Thank you.
Have you tried that? Because that was a question I was going to ask and she just hijacked it.
So those studies, as I said, we have seen that in plasma it is 1%. So even in plasma it's 1%. So if it's plasma, it's 1%, BBB crossing is very, very, very low.
Yeah, but plasma is a dynamic system.
Yeah.
While your brain tissue would be more having like a, what we call accumulation. So
Yes.
if you keep on repeating giving it, it will cross the barrier and hopefully it will accumulate in the brain
Yes.
tissue. So if you do a like a tissue distribution study, you might be able to find it, and that will add a lot of juice to the whole story.
Yeah. So what we are doing is we are doing two things now here. One is estimating the brain concentrations like PV-PK modeling. You might know this. And the second one is to formulate quercetin in a way that my colleagues are doing at NIPA Raebareli, so that we have the evidence that how to formulate in order to make it a blood-brain barrier crosser. So those guys are helping me out in making those formulation. Now that study we will do all together to find out the bioavailability, formulation, quercetin per se, and this, whatever we are talking, the PV-PK.
And any data that you have on quercetin on some sort of very robust disease model of CNS? Because what you showed, those are nice and those are more on behavior.
Yeah.
But how about like a disease model which is induced by a very robust inducer?
We have not checked that.
You haven't checked that.
That we have to do.
All right. Sir has a query.
Yeah.
Just one question. Do you think there's any opportunity that using nanoparticles could increase blood-brain barrier transparency?
Yes. The research shows that using nanoparticles increases blood-brain permeability. But again, like scaffold-based variations are there. Depending upon chemotype, it may behave differently. Like as we see in quercetin, we have several hydroxyl groups over here. So that hydroxyl group makes it somewhat polar, presence of these hydroxyl groups. So that is the main reason for prevention of absorption at GIT and also for this BBB crossing. So we have to think, in addition to nano, how we can formulate it in a way that this facilitates this hindrance of OH group. That we have to see. Maybe some linker or something else which metabolizes in the brain and the drug gets free.
You mentioned a little bit about the mannitol use.
Yes.
Which actually its clinical implication is very significant in head injury, where they want, they're very careful that only the flow is one way, because the trouble with mannitol, trying to increase the blood-brain barrier, which is based on osmosis, can have a reverse osmosis after some time, because then it starts absorbing more into the brain. It will cause, initially, it will help your cause, but later on it will just give away. So, did you try using it, or it was just a hypothesis or a thought?
We tried using it. We used it in this model.
How did it work for you?
We used it intraperitoneally, not in brain directly, not in transnasally. So what we saw in literature also, that people have used intraperitoneally, and they are saying when you give it intraperitoneally, it will come in blood. And for some time it will be in blood, as it is sugar, so it will be metabolized faster.
Exactly.
So it will do its work and go out.
Go off.
Yeah.
Of course, it is given to the peritoneum because of the surface. It has more surface to get absorbed. If you give it directly, it won't work. Before it works, it will be out.
Yeah.
So second part is where you need to be careful that it may actually lead to the reverse flow.
Okay, we'll take care of that.
Yeah, thank you.
Thank you.
So I have one question. This is regarding the transnasal translocation. What is the success rate for this delivery across the transnasal into the brain when you're giving it through the nose?
We are not trying this transnasal delivery for quercetin because we understand, like, for now, if you think of clinical compliance, transnasal, I don't know, maybe clinicians would be a better person to say about compliance of transnasal formulation. So we are using this for developing model of Alzheimer, not for treatment of drug.
Agreed. My main purpose was to know, even when you are delivering the beta amyloid through the nasal chamber, how much is actually getting delivered into the brain? Have you quantified it somehow?
That we have not measured in terms of quantity. That only we have done in terms of histology, which we see here. That's it. Okay. So, and we see whatever is deposited is being cleared off by quercetin, maybe by some mechanism. So this mechanism, we are more interested in how this is taking it down.
And what was the vehicle that you were using for delivery of this beta amyloid? Is it like the pure protein you were delivering?
PBS, phosphate buffer, yeah.
Okay.
Thank you very much, Rakesh. Fantastic talk. Thank you very much indeed.
Thank you very much, sir. I now invite the Dean for Postgraduate Continuing Education and Research School of Dentistry, International Medical University, Malaysia. He has been very active in practicing endodontics and training young dentists for more than 17 years and has a vast experience of microendodontics and retreatment cases. He is the recipient of the Best Faculty Presentation Award in International Association of Dental Research held in San Francisco, USA 2017. He has published more than 80 scientific papers and has authored 5 chapters in 3 textbooks. It's a pleasure to have you here, sir. Thank you.
All right, until he is doing it, so let me start my topic. So first of all, thank you so much, Shri Swamiji and Shri Acharya ji, and Professor Varshney and my dear friend Dr. Kuldeep Singh, he's there, for inviting me here. And honestly, I really made it last minute. So I know all of you are very tired and waiting for me to finish fast. And I would say you can sleep very well now because my lectures are really boring. And I make it more boring as well. So all of you, those who want to sleep, please go ahead, sleep. And so this shows you're really into, yeah? All right. So I am Dr. Abhishek Parolia, working in International Medical University, Kuala Lumpur, Malaysia, as an Associate Dean for Postgraduate Continuing Education. And actually, I was very happy in the morning when Swamiji was addressing the audience. And he addressed me by saying, young scientist. You know, I mean, after a long time, someone told me young, because my wife always keeps telling me, Abhi, you're getting old. And on top of it, the first lecture, which was by Professor Chintamani, I really loved his lecture because when he said you need to be happy. Because when you're happy, your cells are happy. When your cells are happy, you will stay younger forever. So now I'm going to tell my wife that Swamiji told me I'm young and I'm happy, so I'm going to stay young forever. So she cannot say I'm old enough. All right, so this is the place where I work. This is in heart of the city in Kuala Lumpur, which is the capital of Malaysia. And By profession, I'm a clinician with a research background. So this is a clinical setup where my students work, and this is my preclinical setup where my students do preclinical training. And as the theme goes, plants to patients. And as I mentioned to you, I'm sure all of you, those who are here, I was told that you are not dentists. You are better than dentists, right? So all of you are researchers, you guys are into medicine, some of you are in Ayurveda, in yoga, and so many other complementary medicines. But actually I say, dentistry is unique because everything goes through mouth. So if you want to improve your systemic health, you need to improve your oral health. And when I say this, I stand by, because most of the problems what you have in your body can be actually found out by looking into your mouth. So those who brush in the morning with Dant Kranti, because my son, you know, I put him in Hindi classes, and his teacher calls me and tells me, Dr. Parolia, don't pressurize him to learn Hindi. He really understands Hindi, but he can't speak Hindi, but he can speak Dant Kranti. So when I told him I'm going to meet Swamiji, so he's like, the same one who invented Dant Kranti? I said, yes. So he does brush twice daily because I'm a dentist, so I keep telling him to brush well. So I'm a root canal specialist. So those who don't brush well with Dant Kranti, you'd be ready to go to Dr. Kuldeep Singh here. All right. So the topic which I chose, actually the journey started almost like 20 years back. And when I first time I started using propolis. And propolis is something which has really made me think that how natural medicine can help us, even in dentistry. So when I started talking about this, a lot of people didn't even believe me. And they said, oh, it will just one-time event and nothing will happen. So for next 15-20 minutes, I would like to give you a bit of glimpse that what I have done with my team for last so many years. And I hope, I'm not showing you what is the best here. What I want to show you, I want you all to be alert to find faults and tell me these are the flaws here so I can improve on that. So I'm here to seek help from all of you that you as a scientist, a great scientist, can really help us to improve the oral health by doing better inventions. So this is my team from Brazil. And I've been the frequent visitor of Brazil, not for the beaches, actually for the work which I was doing with. And whenever I go to Brazil, I get a lot of information on propolis. And you know, the food is amazing there. After a long time, I had food here in the afternoon, which was served to me like this. I was really mesmerized. So thank you for the hospitality. I really owe you all. So the topic which I chose, propolis, is amazing, actually. It is a honeycomb extract, and which have got a lot of properties. I mean, including anti-cancer properties. And one of the cancer group, they invited me to be the part of their team, but I was not directly involved. But I had given some of my work there, so I have not included that part in my research today. But I am going to show you other things, what I have done in the field of oral health. Now, propolis actually is of three main types, which is brown, green, and red propolis, and there are 12 different botanical origins. The red propolis is very unique because this you find only in the northeastern part of Brazil. You don't get it anywhere else. However, the green and the brown you get everywhere, including India. So, it has got various compounds. As you know, any natural product will have a lot of components in it. So propolis also has more than 200 components and a lot of flavonoids. And we all know flavonoids, like before me, Professor Rakesh Singh was talking about quercetin, which is one of the common components which I found in propolis as well. So let me take you through what we have done in our research. So this was my first research which I actually carried out in 2004 on pulp capping. And it was on humans. So what we were doing, pulp capping is basically, as you know, the human tooth has got the heart of the tooth is the pulp, which has got the nerves and the blood vessels. I'm sure how many of you have had toothache? Once in a while you will have it, right? So when you have a toothache, it's basically because of the nerves which are present in your pulp tissue. And that is a collagenous tissue. So sometimes what we do, we don't go for root canal, we try to preserve the pulp tissue. And there are a couple of commercially available products, which is MTA and Dical. A guy who invented MTA became a billionaire in 3 years' time because that changed the entire scenario. But again, it is nothing. It's like a Birla cement or a Portland cement in the US. So what we did, we compared that with propolis. And what we realized that after 15 days, what you see here, the propolis had created a bit of bridge similar to MTA. And after 45 days, what you see here under histopathological examination sections, that the propolis was really wonderful. As similar to MTA, what you can see. So one thing is there, the MTA is very pricey. We all dentists know MTA is like a gold. 1 gram of MTA is almost like 5,000 rupees in India. So imagine how expensive it is that. So and propolis is like what you get everywhere. So that was very useful. Similarly, just raise your hands. I can see whatever I can. How many of you feel sensitivity when you drink something cold or something hot or something very sourish? I'm sure some of you will be feeling it, right? Those who those who don't want to, please raise your hands. Yeah, I can see, oh, so many. So what do? You go to a dentist and you say, fix it up. And what do? We are very smart people. We provide desensitizing agents. I'm sure all of you must be knowing Sensodyne, Colgate Plus. I'm not mentioning any company by just because they have paid me, no. I'm mentioning because that's how we use. So what did we use? We used red propolis. I got red propolis from Brazil. I got an extraction done. And then what did we do? We did the same thing on the natural teeth. So what you see on the left-hand side is the red propolis, which we compared with Sensodyne, which have got Novamine inside, and Colgate Sensitive, which is commercially available, which is arginine calcium carbonate. And what did we do? We actually did the same experiment on the natural teeth. So we got extracted teeth, we applied these agents, and after applying these agents for 30 seconds, we checked under scanning electron microscopy whether the tubules, which have got nerves inside, if it open, it is blocked. If it is blocked, you won't have the sensitivity. But if it is open, you will have the sensitivity. And now what happens is, even after desensitizing agent has been applied, we wanted to do acid challenge. So acid challenge is basically like you apply desensitizing agent, but then you go back and start drinking Coca-Cola, or start drinking citrusy fruit, or juices, or for example, lemonade. And that will again open up the dentinal tubule. So we want something which can actually resist the acid challenge as well. So it's not only you apply one time and it is going to be effective. It should be effective even after acid challenge. And that's how we realized that the propolis which we used here, this is before application of any agent, when all the tubules were opened up. And once they were opened up, then we applied the agents. And you can see propolis here, and even on the second one, which is Novamine, or the third one, which is calcium carbonate, all were blocked. But what was interesting here is after acid challenge. And you can see after acid challenge, propolis was the only agent which kept your dentinal tubules blocked. So one thing is there, if you are using the propolis in the toothpaste, that is going to have a better relief over Sensodyne or a Colgate Sensitive, which are really commercially available, and here you can see better results. And these are the published results in one of the very reputed journals. So I also wanted to check the effect of propolis in wound healing. And as I mentioned to you, I'm an endodontist, so I work with the heart of the tooth. And heart of the tooth is pulp tissue, and pulp tissue is a collagen tissue. So what did we do? We actually compared the effect of propolis on to the soft tissue, which we actually had given an incision. And we wanted to see the cell migration and proliferation. But here I wanted to compare propolis of different origins, one from Brazil and one from Malaysia. And what I realized, the one which I'm using from Brazil, even at the lower concentration, it can actually help you to improve the cell migration as well as proliferation. So if I show you my next picture, you can see over here, on the top, it is Brazilian propolis, which is working at 10 microgram per ml, and after 24 hours, there was a nice cell maturation, which was completely closing the gap. However, with the Malaysian propolis, we had to use higher concentration. So one thing was very clear. From different geographical location, things will change. So from India, it may change. And to be honest, there have been a lot of papers on Indian propolis, but I find a lot of shortcomings there, which I'm going to show you later. So this is very promising because we want healing. I mean, even in endodontic infection, which goes into the bone, what do we want? We want the healing to occur. And if we are using propolis as a medication inside the tooth, or as an irrigant, which I will be talking in 5 minutes' time, you can actually see the healing happening in the bone tissue because your body host response is amazing. So what did we do? I started thinking in endo. Endo means like, which is my field, endodontics, where you actually clean the canals, you remove the infection, and then you place the medication within the tooth. So when you place the medication within the tooth, we expect all the microorganisms to be completely killed. And these microorganisms are very, very important because that's how we see clinically. As I mentioned to you, I'm a clinician. You can see there is a lot of infection here. And what do? We clean them up, and after cleaning, we fill them up. So you can see the black line here, which is bone if totally gone here, and now the bone is regenerated. So the regeneration of bone will happen once the microorganisms are gone. You can see the same thing in my next slide, where I clean them up, and after cleaning, what you see here is a total bone loss. And then, after a year, it's a total regeneration of the bone. So when you see these kind of effects, the patient is happy because you don't need to take out the tooth, and you can save the tooth. And what we say natural, anything natural is good for us. So this gave me an idea, because how much we try, infection will occur. How much we try, treatment will fail. One of the common reasons for the failure is microorganisms. So everything revolves around how can we reduce the infection. And especially in dentistry, everything revolves around that. And I'm sure in systemic health as well, how to reduce the infection. To be honest, in oral cancers, we have found the role of fusobacterium in the cancer cells. So it is not only the cancer cells, it is even the microorganisms which are very important for us. So, and as we know, there are various microorganisms, and the top 3 microorganisms which we have found out from the literature is E. faecalis, fusobacterium, and P. gingivalis and Propionibacterium nucleatum. So my work is mainly on E. faecalis and fuso right now, which I'm going to show you, but I'm also working on P. gingivalis as well. Now, when I say this, we need to understand the root canals, if not in one dimension. It has got different dimensions, so it doesn't even go in the same vertical dimension, it can go laterally. As you can see all these microorganisms, it can go inside. So what happens is, when you clean the canals, this is the canal what you have. When you clean the canal, you can clean this side, but the microorganisms which have gone really deep inside, it may not be possible for you to clean up. And that is why we need something like drug delivery, which can have a smaller particle size. That is why the nanoparticle came in the picture, which can really go deep inside and can help you to remove the infection from the root so that the healing can occur by itself. And that's how we did. It's against the biofilms. So what did we do? We actually produced chitosan propolis nanoparticles. So we used chitosan as a polymer. And today morning when we were having a discussion, I think Dr. Bhattacharya was giving a lecture, and then suddenly he spoke about carbon dots. And when he spoke about carbon dots, he mentioned that can go up to 10 nanometer. And Dr. Vaishnav was next to me, and I immediately asked him, hey, 10 nanometer, don't you think it will clump? And he said, no, this is the beauty of carbon dot. So this is something which I'm really going to look into, because I use chitosan. And for chitosan, if you go really small, it clumps. But anyway, that is a future direction. So what did we do? We actually produced propolis. We got that extraction done, and we added on chitosan. As you know, chitosan is a polymer, which is a vehicle which will take your propolis along with it. And then what we care, as you all know, because here you are all scientists, I don't need to tell you how nanomedicine is evolving. Nanoparticles are really helping us because it can distribute way well in deeper areas where your regular medication may not go. So everything is going towards nano. Now when I talk, not nano car, nanomedicine. So when it is going towards nanomedicine, so what did we do? We wanted to carry out the experiment as a medication or as an irrigant. Now to simple words, I know you guys are not dentists. So medication is something, imagine if you have a hollow tube of tooth and you put something inside the tube and allow the tube to be disinfected. Or you flush that tube with some irrigant which can allow the tube to be disinfected. So we have to produce something which can clean the tube in such a way that it can allow the healing to occur, which is your natural host response. So we had Malaysian propolis. I got Malaysian propolis and I made the extraction of Malaysian propolis based on alcoholic 1, and you can see the entire one. And we carried out HPLC. When we carried out HPLC to find out the components, the 3 components which were really promising were pinosembrin, quercetin, and quinferol. These were the 3 flavonoids which we found in Malaysian propolis quite regularly. And that's how we started working on. So what did we do? We actually wanted to attach these polymers, like chitosan polymers, and we attach these polymers, propolis on these polymers so that it can take my propolis as a bioactive molecule deep inside the tube which I have spoken about. Once it was done, then we also had to characterize the nanoparticles, what we had produced. And you can see the nanoparticles were almost like 100 nanometer, 10 to 100 nanometer to be exact here, which has high zeta potential and high and spherical in shape. So what did we do? We actually had the tooth. So I mean, I'm just giving an example here. So we had multiple teeth to ensure, and I created the biofilm inside. So we inoculated the E. faecalis broth inside the teeth. And thereafter, we compared these medications. So if you look at the CH, which is calcium hydroxide, and if you look at this propolis and the chlorhexidine, these are commercially available ones, which people have been using for ages. And whether it is good or not, we are still using it without even realizing that we may find something better. So what did we do? We actually compared these medication, and we wanted to see whether chitosan alone can work also or not. And what we realized that within a day, because I compared for 3 days, 1, 3, and 7 days, within a day, my chitosan propolis nanoparticle, which was higher in concentration, 250 microgram per ml, it actually eradicated all my biofilms within a day, which was way better than which we already have in the market for so many years. And not only to 200 micrometer laterally, it also went quite deep. So you can see it can really go 0.5 millimeter, which is quite a lot. Imagine if I'm cleaning this room vertically without cleaning sides. So what will happen? I'm leaving a lot of microorganisms on my sides. So I want these nanoparticles which can go vertically, which can go laterally, and clean up the entire room so that the healing can occur. And definitely we had to ensure it is working. So we cut the tooth and we did the scanning electron microscopy. And what we realized, we wanted to see whether this entire tooth is covering with the E. faecalis microorganisms. Or what we realized here, the chitosan propolis nanoparticles, if you look at this one, the 250, it was absolutely clean after 1, 3, and 7 days. However, the calcium hydroxide one, which is really there in the market for more than 50 years, after 1 or 3 days, it was not able to eradicate. So one thing is very clear. The one which we have been using, we have to rethink. And what goes here? Rethink ethnopharmacology. So I think we all need to revamp and rethink and reevaluate, are we doing enough? And are we going in the right direction? So we also went for confocal laser microscopy. So I was privileged to see the center where I could see the facilities, amazing world-class facility. I must tell you, Acharya ji, it was really, really good. Okay, and yes. I mean, I have seen the facilities. I was giving a lecture in Harvard just 2 months back, and I tell you, it was that level. So it was really amazing. And what I realized that in the cone photo laser scanning microscopy when we did, we found out that the CPN, which is chitosan propolis nanoparticles at this concentration, completely eradicated these microorganisms. So that gave me a chance to see, okay, let me try from the patients then. And then what did we do? We collected the samples from the patients. We got the samples from the patient, we got the microorganisms from the infected root canals, and then after collecting from the patients, we found out the CPN 250 was again very effective, even from the samples which were not from the laboratory, from the patients. So that gave me an insight, okay, so I need to use CPN, but this is against one microorganisms. I will show you for other microorganisms as well. And that's how we carried out the entire experiment. But we also wanted something to flush away the infection. So medication we placed there for a day or 3 days or 7 days, but the water or any cleansing solution we need to clean this room, we need hand in hand. So it has to be effective in minutes. So we had prepared nanopropolis. And nanopropolis, what we prepared, actually we prepared by ultrasonication. And then after preparing the nanopropolis, we actually got the characterization of propolis, as I mentioned to you before. And once we got it, the most of the nanoparticles were in the range of 10 to 100 nanometer. And I'll be very happy if my particle size goes down less than 10. But again, we were having the issue of clumping, which I hope we can work on that later. And this was, all the particles were almost like spherical in shape, which shows that it has got a better shape than irregular shape. So what did we do? Here we compared with, again, commercially available, and one of the very, very toxic irrigant, which is called sodium hypochlorite. I'm sure all of you must have cleaned your floor once in your lifetime, unless you are a minister. Right, ministers don't do. Right, all of us need to clean the floor. And if you clean the floor, you get Clorox. Clorox is a floor cleaner. So similarly, Clorox, we clean the canal using Clorox. The difference is they use 100%, we use 3 to 5%. That is the only difference. Imagine you are using in patient's oral cavity. And when you use that, there is a lot of problem as well. So I wanted something which can replace Clorox. So we compared this with nanopropolis, and what we realized, so you can see different components of propolis as well as Clorox and chlorhexidine. And what we realized that after 1 minute, it was not very effective. After 5 minutes also, it was not very effective. But after 10 minutes, it was as effective as Clorox. So now when it is as effective as Clorox at 200 micrometer as well as at 400 micrometer deep, one thing is very clear, that I can try this irrigant, which is having the same antimicrobial property as Clorox without having the toxic potential of Clorox. So that gave me a bit of insight that, okay, let me try this more. So what did we do? So we did SEM, and you can see in scan electron microscopy, if you look at the third from the left, when I'm using the propolis nanoparticles at 300 microgram per ml, it was as beautiful as sodium hypochlorite, what you can see the next to it on the right. So we went for cone photolaser scanning microscopy as well, and we realized that the complete eradication was possible after 10 minutes when we compared with 6% sodium hypochlorite compared to 300 microgram per ml of propolis. So propolis, which is natural, which we can consume, is way better than Clorox, which we can't even consume. So we also did the spectroscopy, and we realized you can see the projections, and we could say that it is really killing the microorganisms at that level. As I mentioned to you, we also went for transmission electron microscopy to actually see where exactly it is affecting. And we could realize that it is actually breaking the membrane and letting all the cytoplasmic organelles to come out of that and killing the microorganisms. You can see it here. So you can see it was totally off, then it will kill the microorganisms completely. As I mentioned to you, E. faecalis is not the only microorganisms which is responsible, even the fusobacterium, which is very, very difficult to kill and remove, especially from the root canals, we found that even for fusobacterium, it was quite effective, and it could kill the fusobacterium, you can see by transmission electron microscopy images as well. We further went ahead with confocal microscopy, and we could see it was totally removed. You can see this. And compared to E. fetalis, even the fusobacterium was able, it was able to remove that as well. Now I wanted to do something more molecular, because that is my problem. I don't get happy so fast. So if I find something, then I have to find something else. And that has been a problem all the time. My wife doesn't like it. Yeah, she always tells me, you're never rested. Yesterday when I came in the night, I went to the gym. So I told my wife, I'm going to the gym. She said, you know what? You were working until Sunday, you are flying on Tuesday, all the way you flew, and then you want to go to the gym. Don't do that. But you know, I always have a tendency to stretch myself a bit more. So I wanted to do the same thing with the molecular docking, where we wanted to see, because you know, E. fetalis have got galactosidase and sortase A. And we have realized there are three main flavonoids, which is timferol, pinosembrin, and quercetin. And we wanted to see whether it is really attaching on that, and there is an attachment. What we realized that, if you look at these three flavonoids, actually there was an attachment there. And this attachment molecular docking shows that you can actually kill the microorganisms at the sortase A level, and you can see the beta-galactosidase as well. So this actually confirmed our work that not only at the CFU or scanning electron microscopy or CLSM, we are also able to produce by molecular docking as well. And that confirms that the treatment what we are going to carry out with propolis has got a lot of bright future. So that gave me, as I mentioned to you, I like to be always on toes. So I wanted to see how much data has been published, and that is something I told you about Indian propolis as well. How much data has been published, and what is the quality of papers we are having into the literature. And what we realized that there are a lot of papers have been published against a lot of microorganisms in the various fields of dentistry, starting from endo, from the gum disease, and everything. And I wanted to see, and this is something which those who are working on the natural product, please try to understand, we really need to address the geographical location. What is the type of plant we are using? What is the extraction method? What is the characterization of the material what we are going to use? Are we doing the randomized process properly? And on top of it, are we verifying the presence of microorganisms, especially those who are doing the microbiology work? And you know, when I did all this, I published a paper on gum diseases, where the use of propolis, how can it help in your gum disease by adding into your mouthwashes, by adding into your dentifrices, and it can really do wonders. After conclusion, I realized that there are various differentiating factors which can affect the outcome. That includes your location, the season, concentration, type of plant, extraction method, and the composition. Finally, there is a heterogeneous data in the literature. When you read a data, or when you present the data, I think it is our responsibility to ensure that we are evidence-based and we actually look in the data carefully before we say this is really working. So with this, I would like to end my presentation. Let's try to be more factful than fantasize things. Thank you so much, every one of you, for listening at 5:30 in the evening. Thank you. And these are my email IDs, so if anyone wants to be in touch with me, I'll be very happy. This is something this is my last thing I want to say. I think develop a passion for lifelong learning. And Dr. Vaishnav was sitting next to me in the morning, and then he was talking to Acharya ji. And he said something, I overheard, sorry I'm quoting you. I overheard, he said, I want to do this. And Acharya ji said, you just do. Don't worry about anything. You just do the work. And you know what did he say? I love when the passion meets the nation. Right? Something like that he said. When passion meets the nation, I love it. So I think then I realized you are very passionate, Dr. Vaishnav. So, Acharya ji and Swami ji, and that is why we can see all this here. I think for me, the passionate means simple, is you need to be positive, you need to be ambitious. If you're not ambitious, you can never achieve, but don't be over-ambitious. Be sincere, be scientific, be innovative, be outcome-based, because any research which dies in the lab, it has no use. It has to come to the clinic. Clinic people have to tell you what is happening in the clinic, what are the shortcomings, and then it goes back to the lab, so there is a proper translational research. Be nimble, be adventurous, be trustworthy, and the most important thing, which I believe in, which be ethical. So with this is my, one of the dental magazine who featured me on their cover page and took my interview. So thank you once and all. As I say, let's clean better, and you will have a brighter future. Thank you once again. Thank you.
Dr. Abhishek, thank you very much. This was really, really awakening talk. And I'm sure all the folks who are sitting here in the audience, if they were choosing to take a nap, they actually think it otherwise. So you woke them up, you inspired the thought, and you also made us realize that there are science beyond the body as well. There is science beyond the oral cavity, and there's a deep science in the tooth as well.
Thank you.
So personally, I feel inspired, and I would like to explore more and more and try to go as deep as possible in the tooth, in the close collaboration with Kuldeep and with you, of course. Thank you very much for having this evening and telling us what can be done and what has been done and what lies in the future. I appreciate it.
Thank you so much. Thank you, everyone.
Thank you so much, sir, for your enriching talk.
Om, sir.
Excuse me.
Hello. Excuse me. Sir. Here. Sir, can I ask Hindi? So, मेरा ये क्वेश्चन है कि जैसे मतलब मेरा एक डेंटल इशू है कि मेरा जब कोई ठंडा चीज़ खाता हूँ तो सेंसिटिविटी देन फीवर आता है, उसके बाद सेंसिटिविटी से फीवर आता है. सो ये कैसे ठीक कर सकते हैं सर हम एज होम, ये ज़रा बता सकते.
This is a very good question, actually. You know, for this question I charge. All right. No, but let me tell you, if you are having sensitivity, I mean, definitely, if you say you get fever, which is very unlikely to happen. But if it is happening, you know who is sitting there. Oh, where is he now? Yeah, here. I think you need to see him. He showed me the dental clinic. So please, Kuldeep, you have to see him once, and then let him see.
Abhishek ji, thoda Hindi mein bol do.
Acha. नहीं बोलता हूँ आपको देखना चाहिए उनको और उनसे मिलना चाहिए, ठीक है, और आप मिलेंगे तो आई थिंक वो ठीक होएगा आपका दांत, है ना. और सेंसिटिविटी के बाद फीवर आना थोड़ा अनलाइकली है, मतलब होता नहीं है ऐसा नॉर्मली, तो होपफुली कि आपको ठीक होएगा. तो कुलदीप आप प्लीज देखो उनको, एंड इफ ही चार्जेस ना आपको चार्ज करे तो आप मुझे बताना है. Thank you so much. Yeah. Thank you.
Thank you so much, sir, for your enriching talk on the matter. It was a great privilege to hear you. Now I would like to invite Dr. Kuldeep Singh ji to the dais to present with integration of modern dental sciences. Dr. Singh is presently the dental surgeon and the head of the dental clinic and research center of Patanjali Ayurvedic Hospital, Haridwar. Since September 2006, he is an active member of the Academy of Oral Implantology, Dentist Association of India, Indian Dental Association, and the American Academy of Implant. Dr. Singh has published 12 research articles in 12 papers published in various national and international journals. Please carry on with your presentation, sir.
Thank you very much. So, Param Pujya Acharya ji, महाराज एवं स्वामी जी महाराज के चरणों में सादर चरण स्पर्श एवं यहाँ पर आए हुए सभी विशिष्ट अतिथियों को मेरा प्रणाम. जैसा कि हम देख रहे हैं कि as we know that the today's topic for discussion is that integration of traditional Indian knowledge of yoga and Ayurveda with the modern dental science. So when we think about the yoga and Ayurveda in dental care, we generally think that it's a preventable thing. It can prevent the disease, but how we can use yoga and Ayurveda in different dental diseases. So, sir, now the scenario has been changed. Now, even in the previous presentation, we have seen that the use of propolis as an integral part of dental treatments, during the use of root canal treatments, as an irrigant, as a pulp capping, and so many more. So, the relationship between our traditional science and the modern dental care is like that. We can describe in one line that treatment ke saath bhi or treatment ke baad bhi. So it's a no more a preventable disease, but we can use yoga and Ayurveda as a supportive dental treatment therapies and preventive as well. Sir, as we know, the oral diseases continue to be the major health problem worldwide. The most common dental problems are the dental caries, gingivitis, and periodontal diseases. And it also affects the economy of the patients, as the total 10% of the health expenditure only goes to the dental care. So we can use our traditional knowledge of herbs and yoga practices may apply to the holistic approach of the dental treatment and prevention of the oral diseases. About 34% of the tooth decay goes untreated and results in the different kinds of the dental problems like pain and discomfort, etc. These are the common impact factor of the oral diseases on the daily life, like the fear and anxiety, pain, discomfort, impaired growth in the children, impact on the general health, treatment cost, sleepless nights, time of work, reduced productivity of the workforce due to the pain. Sir, the common factor affecting the incidence of the dental caries or dental diseases. There are two kinds of the factor that affect the incidence of the dental diseases, like the local factors and the systemic factors. I'm not discussing much about the local factors here, as it is a well-established facts that what is the reason behind the dental caries and the periodontal diseases. But I just want to discuss about the some systemic factors like stress. Sir, when we are planning the, doing the diagnosis and the treatment planning about the dental diseases, we generally don't consider the systemic factor like stress. We only concentrating upon the local factors that are present, and we do the treatment for that one. So I want to explain you that how the stress can create most of or all of the dental diseases alone, even in the absence of minimum etiological factors. The stress can either increase the incidence of the dental caries or aggravate the pre-existing dental diseases. Stress can also compromise the ongoing dental treatment, their successfulness. So we have to consider the stress as one of the important etiological or the affecting factor in dental diseases. Now I will explain how the stress can affect or can increase the incidence of the dental diseases. Sir, the first of all, as we know, the tooth brushing or mechanical plaque control, what we say, is a motivational based process. And when we are stressed or we are in a persistent stressful conditions, we lack the motivation for the tooth brushing and hence aggravating the pre-existing dental problems. Second, the main cause is the exaggerated inflammatory response of the gingiva. Because of the persistent stress or long-term stress, there is increased secretion of the cortisone, and hence it results in the increased plasma levels of the pro-inflammatory cytokinins, that is TNF-alpha and IL-6. The stress also causes the decreased level of the IgA and resulting in the immunocompromised condition, and altogether leads to the increased fluid accumulation in the gingiva. And long-term fluid accumulation in the gingiva leads to the anaerobic condition, and that can aggravate the pre-existing gingival problem or the periodontal problem, and hence resulting in the bleeding gums and foul smell. So the exaggerated inflammatory response aggravates the pre-existing gingival or periodontal problems. The third most common factor is when we are under the persistent stress, there is a decrease in the salivary secretion. And hence, as we know, the saliva is the self-capacity of the oral cavity to clean. So, in the condition where when we have a decreased salivary secretion, there will be less cleansing of the oral cavity, and hence resulting in the more incidence of the dental caries, as well as the increase in the periodontal disease symptoms like the bleeding gums and the foul smell. The fifth most common factor that affected by the stress is teeth grinding. It's a body general phenomena. When we are under the persistent stress, we just release it out by some way, and the teeth grinding is one of the way. And when we have a persistent teeth grinding, that results into the attrition of the teeth, that is the layering off of the enamel, and hence resulting in the opening of the dentinal tubules and resulting in the sensitivity to the teeth. It also leads to the micro cracks in the tooth and the fracturing of the broken fillings. Sir, constant, when we have excessive teeth grinding, it results in the TMJ pain and TMJ derangements. Under the constant stress, sir, when we keep grinding our teeth, it results in the overworked jaw muscles, and hence resulting in the myofascial pain dysfunction syndrome, where we have a constant pain or headache or pain in the facial muscles. So these are the main factors that are affected by the stress. Even in the absence of a proper etiological factors, we may have all kinds of the problems. So, sir, during the diagnosis and the treatment planning of the dental diseases, we must consider both the local factors as well as the systemic factor for the long-term successful dental treatments. Sometimes, in the absence of local factors, the stress or the other factors could be the main cause of the disease. Like sometimes we have a quite good oral hygiene. There's a minimum deposits in the mouth, but still we have a foul smell or bleeding from the gums. That is because of the exaggerated inflammatory response of the gingiva, which results in the foul smell and bleeding gums. So the treatment should address the problem, systemic problems as well as the local problems. Now I'll just little discuss about the integrated treatment approach methodology. Sir, the dental caries. As we all know, the dental caries is a chronic multifactorial disease known as the tooth decay or tooth cavities. It is the most common non-communicable disease and a serious public health issue in the world. Caries causes the destruction and demineralization of the hard tissue of the teeth by the acid production occurring from the bacterial fermentation of the food. Sir, the common treatment factors for the dental caries are, there are two kinds of the treatment. One is the external mode and second is the internal mode. External mode may have like a filling, a root canal treatment, fluoride applications, crown, bridge, partial prosthesis, etc. But again, the internal mode, because the disease is created by the bacteria, or main factor is the bacteria, so we can use some antibiotics like erythromycin or penicillin are the popular antibiotics. But recently we have seen that the resistance has been found in the cariogenic bacteria. So the bacterial resistance and the side effect of antibiotics make interest towards the natural products. Moreover, some plants have shown potential against the dental caries, and some of these are like, as mentioned here, the jamun, satut, and neem. These are the known plants extract that have the cariogenic effect, anti-cariogenic effect. Sir, idea to say this plant extract is that we should work on that to prepare some paste or mouthwash that has herbal anti-cariogenic extracts that can be used in the children's for a mass prevention of the dental caries. There is more research as well as the product development is required in that area, because as we have a fluoride application in general for the children's to prevent the incidence of the dental caries. So instead of, because fluoride has their side effects, excess fluoride has the side effects, but these plant extract doesn't have any side effects, and they are known to have anti-cariogenic effect. So our research as well as the product development must concentrate on, or must have a line to develop a product which can prevent the dental caries in children. Sir, I will just say a few words about the pulpal diseases. As sir has told in the previous presentation that when the dental caries reaches up to the pulp, that is the pulpal problem. And as per the severity, it can be described as the irreversible pulpitis, apical periodontitis, or periapical abscess. The treatment of choice for any one of these problems is the root canal treatment. And during the root canal treatment, we use antibiotics and painkiller as a supportive medication. The root canal is a standard treatment, but to overcome the symptoms of the pain, we have to use the painkillers. And the most commonly used painkillers are the acyclovir or NSAIDs. And the main problem with these painkillers is their side effects. Any long-term use of any of these painkillers may develop a gastric irritation or allergy. So there is a need to develop a herbal painkiller with no or minimum side effects so that it can be used. Sir, it's proud to say that our institution has developed a herbal painkiller named as the Perennial Gold and is now available in the market after a successful clinical assessment in dental pain and the pulpal diseases. So we carried out a small clinical research work on the Perennial Gold and the endodontic pain. That is the efficacy evaluation of traditional Ayurvedic formulation Perennial Gold on the pain intensity management of endodontic pain, a randomized controlled clinical trial. And the result of this study was, it was found that this was a non-superiority study, and the outcome explains that both the groups depicted the similar effect in managing the endodontic pain. So, the result of this study is that we can use Perennial Gold as a substitute to any NSAIDs in endodontic pain or any other dental pains, like a periodontal pain or endodontic pain, without any side effects. Thank you. So there's one more article relating to the yoga, sir. Because the pain is one of the main concerns in dental care, whenever we do the treatment, there might be a painful condition. So as a herbal intervention, we can use Perennial Gold. But yoga is also known to increase the pain tolerance capacity of the individual. So this was a small clinical trial we carried out in the patient suffering from the dental pain and their pain tolerance. So the title of the study was the increased pain tolerance in dental patients by yoga and special reference to the pranayama and bhakti yoga. And as a result of this study, we found that the patients who are regularly practicing the yoga have increased pain tolerance as compared to the patients who are not practicing the yoga. So it can be said yoga is useful in increasing the pain tolerance, especially even in the dental patients. Sir, the periodontal diseases or the pyorrhea, we can say both the pyorrhea or the periodontal diseases. Out of 10, the 4 patients are suffering from the periodontal diseases or the pyorrhea. It is the infections and inflammation of the gingiva and the surrounding connective tissue to the bacterial or plaque accumulation on the teeth. It's a bacterial infection. The main bacteria involved are the P. gingivalis, P. intermedia, P. denticola, etc. The main common symptoms for the periodontal diseases or the pyorrhea is the bad breath, bad taste, red swollen gums, tender or bleeding gums, painful chewings, loose teeth, sensitive teeth. The most common symptom is the bleeding gums and the foul smells. That I think everybody suffers from that. And that is the inflammatory response caused by the periodontal disease can complicate already existing diseases such as the diabetes, heart diseases, and the kidney diseases. So, it has been also found, sir, that the bacteria related to the poor periodontal health are also responsible for the heart problem known as the infective endocarditis, which may result in the heart problems or heart failure. So the good periodontal health is related to your good heart as well. Or if you have poor periodontal conditions, that may lead to the heart problems known as the infective endocarditis, and that is a well-established fact. So there is an integrated approach for the treatment of the periodontal diseases or the pyorrhea. First of all, there are the professional treatments or the dental treatments that are required, because the main cause of the disease is the food impaction between the teeth and in the pocket. So we have to clean that local factors as a primary treatment. So the oral prophylaxis or scaling is one of the best procedures or common procedure we do for the periodontal diseases or the scaling, and it is not a harmful procedure at all. Another procedure we can do as per the severity of the disease is the gingivectomy, root planing, gingival curettage, etc. As a supportive therapies, we can toothbrushing with the toothpaste, interdental cleaning to clean in between the teeth, mouth rinses, gum massage, and yoga intervention. I will tell you about the oral prophylaxis. That is the procedure. Scaling is the basic procedure required to clean all food impaction in pockets and interdental areas. During this procedure, the patient has mild pain and discomfort. To manage this, we have to use either anesthesia or patient has to tolerate that. So here in Patanjali, sir, we have a herbal desensitizer known as, which has been used as a local anesthetic substitute known as the Divya Dhara. So, Divya Dhara as a desensitizer before doing supragingival scaling. This was the small study that we carried out in our dental patients undergoing the cleaning procedure or the oral prophylaxis. The title of the study is The Effect of Divya Dhara, a polyherbal formulation on the assessment of pain and discomfort during supragingival calculus removal, a clinical study. The design of the study was a double-blind, placebo-controlled, randomized trial. Assessment, the pain levels were assessed by after using a visual analog scale, that is the VAS. And the effect of this study was that the Divya Dhara is significantly reducing the pain and discomfort during the supragingival scaling. So this is the herbal intervention that we can use as a supportive treatment methodology in the case of, along with modern dental treatments. So with the use of the Divya Dhara, we can avoid the use of local anesthetics that are required for this procedure. Thank you. Now I'll just emphasize on the brushing and the toothpaste. Sir, as the periodontal diseases are paria, the mechanical plaque control is one of the best way to control the disease. What does it mean by the mechanical plaque control? That is the toothbrushing that we do. So, in the toothpaste, we generally use synthetic substances like triclosan and chlorhexidine, and they have been found to have some side effects like staining of the teeth and burning sensation. So, this led to the paying increased attention on using natural ingredients in herbal dentifrices. Commonly used herbal ingredients are the akarkara, neem, babul, haldi, etc. The herbal ingredients are known to have antibacterial as well as the anti-inflammatory properties. So this is one of the studies that we did on our popular toothpaste, the Dantkanti Natural. This is a comparative study between a herbal toothpaste, Dantkanti, and the non-herbal toothpaste. The study was designed as a two-group comparative study. 50 subjects are in each group. The indices examined are the OHIS and the gingival bleeding index. The conclusion of the study is that the herbal Dantkanti toothpaste is more efficient in maintenance of the oral hygiene and gum bleeding as compared to the non-herbal toothpaste. So it's always better to use a herbal toothpaste instead of having a normal toothpaste, because they act on the both cleaning capacity as well as the antibacterial efficacy. Another main important factor of the herbal toothpaste is the antibacterial efficacy. So we did a small trial on that. Antimicrobial efficacy of herbal toothpaste on oral microflora in pre-post toothbrushing study. In this study, we used herbal toothpaste Dantkanti as an intervention. Before brushing and after brushing, the salivary swabs were taken for a microbiological assessment. And the study shows that the brushing with the toothpaste significantly reduced the bacterial and the fungal counts at the 30 minutes interval after the brushing as compared to the control. The article is in the process of the publication. Sir, the mouth rinses. Mouth rinses are the integral part of the oral hygiene. The main components used in the mouthwash is the triclosan and the chlorhexidine. So, because these substances are known to have side effects like the tooth staining, altered tooth taste, and burning sensation of the mucosa. So, the herbal mouthwashes are the need of the time for the best results with the minimum or no side effects. I will little tell you about the Gandusa and Kavala or oil pulling. As described in the Ayurveda, ancient books of Ayurveda, that the Kavala or Gandusa is one of the most important procedures that we do to maintain our oral hygiene. Mouth rinses must be derived from there. The filling the mouth with the morning with the herbal oil or medicated water daily and holding for a few minutes without moving it inside the oral cavity is called Gandusa. Sesame, mustard oil, or sunflower oil or til oil are the important ingredients of this. There are many benefits of doing Gandusa, so I will not go one by one. So I will just tell you that our institution is working to develop the medicated oil and liquid for the oil pulling according to the diseases, like for periodontal diseases or paria, soft tissue disorders like lichen planus, leukoplakia, and oral submucous fibrosis. So we can develop a different liquid for the different disease regime, and they can practice to prevent the disease as well as to treat the disease. So this is the use of Gandusa or Kavala. Gum massage. So this is one of the most important procedure because the gum massage stimulates the gingival blood supply. Natural substances present in the manjans or dant manjans, they have their known antibacterial efficacy. So as we are doing some research work on the Dant Kanti natural has to be used, or we are using it as a replacement of the gum pens. So we can do gum massage with the Dant manjan, but gum massage is not the replacement for the toothbrushing. It's always along with the toothbrush. Like you have to brush twice daily with the paste and brush. You have to do the massage at the end of the day with a small round finger like that. So never replace the gum massage with the toothbrushing. That is the wrong fact, because gum massage is not the cleaning of the tooth, it's the massaging of the gums. And the toothbrushing is the cleaning of the teeth. So the both has to be done together. Now I'll just tell you about the effect of yoga on oral health. Because there are a lot of visitors coming to us and they ask, what is the effect of yoga on dental care? Sir, there are effects of yoga on dental care. As we know, this is the published article that we have. The yoga practices are found to be effective in the prevention of dental diseases, maintenance of the oral hygiene, improving the lifestyle, reducing the stress, and reducing the gingival inflammation. This is one more published article related to the, this was the clinical article related to the effect of yoga on gingival bleeding. And it has been found is quite effective. The only yoga even is effective in reducing the gingival bleeding. Now I will tell how the yoga affects the oral health. These are the some factors how yoga helps in dental care. Sir, as we know that the yoga is known for reducing the stresses, reduce the oxidative level of the body, and hence also yoga is known to increase the motivation. And as we know that mechanical plaque control or toothbrushing is a motivation-based process. So when we do regular practice of yoga, definitely we have more motivation for ourselves, and hence we improve the toothbrushing techniques and toothbrushing. Then again, as we know, the yoga has to normalize the inflammatory markers. Regular practice of yoga, not the 1 or 2 days practice. A regular practice of yoga normalizes the exaggerated response of the gingival inflammation that created by the stress. Because yoga is known to reduce the stress, and hence it has effect on the reducing the exaggerated inflammatory response, and hence it controls the gingival bleeding. It is also known that the stressful condition, exaggerated inflammatory response is hampered with the wound healing. So when we regularly practice the yoga, we have reduced level of the stress, and hence it helps in the wound healing by normalizing the body inflammatory response. It's nothing else, just we are normalizing the body inflammatory response and mainly getting away from the stress. And hence whatever the side effect of the stresses are there in the oral cavity, we can improve all of them. As we are reducing the stress, so we can improve the salivary secretion, and hence we can normalize or we can decrease the incidence of the dental caries as well as the symptoms of the periodontal disease or the pariah. Yoga is also known to reduce the osteoclastic activity and increase the bone mineral density, and hence that is a paramount important in the case of like we are doing dental implants, where the bone is of paramount important. So yoga is known to reduce the osteoclastic activity and increase the bone mineral density. Sir, here we can say that yoga and Ayurveda together reduce the periodontal induced infective endocarditis. A healthy gingiva can have a healthy heart. But if any problem created by the periodontal disease to the heart, it can be reduced by the regular practice of yoga and use of the Ayurvedic interventions. So these are the just. This is one more article that we published. This is the perceived oral benefits of yoga, because till now there is no way mechanism how to quantify the effect of yoga on oral health. So this is the scale development work we did to quantify the effect, perceived benefit of yoga on oral health. And with the help of this scale, we can quantify that what yoga practices are helping us to what level of the oral health. So there is one more article in that line is under process of the publications. So sir, from this presentation taken together, we could summarize that along with the modern dental treatment, integration of our traditional knowledge of yoga and Ayurveda is essential for long-term good oral health and prevention of dental diseases. I would like to thank my team members who have significantly contributed to my work that I have discussed today. And of course, I am blessed with the divine blessings of Param Satdev Swami Ji Maharaj evam Puji Acharya Ji Maharaj that has made me go this far. Thank you very much.
Dr. Kuldeep, this is.
अरे आज डॉक्टर कुलदीप का पहला भाषण था भाई, एक बार जोरदार ताली बजा दो। और बहुत अच्छा अभ्यास किया, और पहली बार कितना साल हो गया हमारे पास? 17 वर्ष हो गया, तो यहाँ सेवा दे रहे हैं, इसके लिए बहुत बहुत बधाइयाँ।
Invite upon stage Mr. Sandeep Sinha Ji for his talk on novel Ayurvedic approach to combat organic and iatrogenic kidney disorders, a preclinical perspective. Sandeep Sinha Ji is currently working as a lead scientist in the in vivo biology department, drug discovery and development division at Patanjali Research Institute.
Om. I'll try to be as brief as possible. Om Ajnanatimirandhasya Jnananjan Shlakaya Chakshurunmilitam Yena Tasmai Shri Guruve Namah. So I bow to the lotus feet of Param Shraddha Acharya Ji, and I also bow to the subtle presence of Yog Rishi Param Pujya Swami Ramdev Ji, who is physically not present with with us today. So I'll be just presenting the preclinical profile of two novel proprietary Ayurvedic drugs which have been developed by PRI, and they are intended to combat the organic and iatrogenic kidney disorders. So kidney, as all of you know, it plays an important role in eliminating the toxins, and secondly, it also releases an hormone known as erythropoietin, which basically stimulates the bone marrow to produce RBCs. So if there are any kidney disorders like acute and chronic kidney disorders, it will basically lead to accumulation of toxins in the body, as well as it will lead to anemia. So what is the role of Ayurveda in combating the kidney disorders? Ayurvedic approaches may include the standalone therapy with herbal medicines yogasan, pranayam, medication, detoxification, and dietary interventions. So for that, today I'll be speaking about a drug, Lithome, that is intended to treat the kidney stones, which is technically known as nephrolithiasis. Yogasan, pranayam, and meditation, they also have a great role. Ushtrasan, Mandukasan, and Mayurasan, and Dhanurasan, they are very beneficial in addressing the distress which is associated with kidney stones. For pranayam, Kapalbhati has been shown to be beneficial in kidney stones. For detoxification, it is said that if there are kidney stones और एक बार आदमी पंचकर्म ट्रीटमेंट करवा ले तो उसे kidney stones recur होने की संभावना बहुत कम रहती है. Dietary interventions are there. Another is the integrative approach with modern drugs, the synergy with modern drugs, improving the bioavailability and mitigation of the undesirable adverse effects associated with the modern drugs. So my second part of the talk will be on vancomycin, which is an antibiotic-induced acute kidney injury. So background, if I give of nephrolithiasis, the earliest reference which you get for kidney stones is Atharvaveda, about 3 to 4 millennia back, and Acharya Shushrut categorizes this as an Ashmari disease, where Ash means stone and Ari means enemy. So a stone which is inimical to your body is known as Ashmari. The main problem with kidney stones is that the recurrence rate is about 50% within 10 years of first detection. So if I go to the brief pathophysiology, it is showing that genetic predisposition, lifestyle factors may lead to crystal formation and growth of kidney stones. There is cell membrane injury due to accumulation of these calcium oxalate crystals and leading to apoptotic cascades. And there is deposition of these oxalate crystals on the apical membrane surface. They enlarge and then they block the flow of the urine. So current treatment modalities include diuretics and non-invasive procedures and invasive surgery for large kidney stones. But they are fraught with their disadvantages, as everybody knows. This is a cartoon showing the kidney stone. The symptoms are enlisted over here. Now coming to the novel herbal mineral medicine for kidney stones. Lithome, this table shows the composition. Gokru is a diuretic drug. Varun is a diuretic drug. Pashan Bhed, the very name signifies that it leads to the dissolution of kidney stones. And we have the mineral components, these various bhasmas and shars. They help in maintaining the alkalinity of urine so that they are dissolved and expelled out of the urine. This is the phytometabolite analysis of Lithome. The key point here being that Lithome is enriched with phytoconstituents which are known to possess anti-nephrolithiatic, diuretic, and antioxidant activities. This compound, Bhajan, which is present in Pashan Bhed, has found to reduce the formation of crystal stones and even break the smaller stones. So this is the first in vitro study which we have performed, the team in vitro biology has performed. So here calcium oxalate crystals monohydrate was synthesized by mixing the calcium chloride and sodium oxalate in a ratio of 1:1. And as you can see over here with this graph, our drug reduced the COM crystal synthesis in vivo, reduced the size of COM crystals in a dose-dependent fashion, and altered the morphology of crystals from sharp edges to the round, blunt edges. Since the COM crystals, they have sharp edges, they have a tendency to adhere to the epithelial cells of epithelial cells. But what lithome is doing is it is basically altering the morphology of crystals from sharp edges to round blunt edges, so that they cannot adhere and they can pass along with the urine. And same data, we did a scanning electron microscopic analysis, and which shows that the calcium oxalate monohydrate had converted to spherical shape calcium oxalate dihydrate crystals, and lithome acted in a concentration dependent manner. So this in vivo model, what we did is we created a hyperoxaluria model, which basically led to the accumulation of oxalate and led to the development of renal stones. So we have taken the male SD rats and we have administered NH4Cl ammonium chloride for 3 days and 0.75% ethylene glycol for 14 days. So after that, we checked the urine for the presence of stones, and once we got the confirmation of COM crystals, we started treating them with lithome at the dose of 40 and 120 mg approximately. And on day 28, we terminated the experiment, performed crystalluria, oxalate citrate analysis.
Can you go much faster on this? Because methods are very easy. If you go point by point, it might take longer.
So this slide basically shows, see, these are the calcium oxalate crystals which are present in the disease control group, and lithome basically it reduced the ethylene glycol induced crystalluria, urinary oxalate, and diminished the and it restored the diminished citrate levels. Going to the next slide fast, oxidative stress is involved in kidney stones, and lithome was able to dose dependently decrease the oxidative parameters and could increase the antioxidant parameters. This is histopathology. This is the normal control group animals, and this is the disease control group animals, where you can see the crystal deposition over here, and you can see that lithome at a human dose, equivalent dose, the crystal deposition has totally gone. So this is this showed a dose dependent decrease in the crystal deposition, and apart from that, we have the inflammatory cell influx, which was also decreased by the drug in a dose dependent fashion. So the next, we have done von Cosa staining, which stains the calcium. As you can see, there is dark stain in the disease control group, and the ethylene induced crystal deposition influx was decreased by lithome. We have also done a 28-day repeated dose toxicity study according to OECD test guideline 407, where we have measured different aspects, and we found that lithome was safe up to a dose of 100 milligram per kg per day. So to summarize, the first part is that lithome retarded COM crystallization, converted to COD, reduced crystalluria, decreased oxalate, increased citrate, and decreased the oxidative stress and decreased crystal deposition, and was safe up to a dose of 100 milligram per kg per day. And hence, lithome holds a clinical therapeutic potential for kidney stones and should be evaluated in human subjects. Going to the second part, the vancomycin-induced kidney injury. In vancomycin is a glycopeptide antibiotic which is used for the treatment of MRSA, but it is frequently associated with acute kidney injury, which is known as vancomycin-induced kidney injury. So, Renograte is a herbal medicine which is indicated for the treatment of kidney disorders, and these are the components of the drug. This is the phytometabolites, and Renograte, it possesses phytoconstituents which are known to have antioxidant and anti-inflammatory activities. So, in vitro evaluation of Renograte on Wiki, we have the in vitro team has basically you generated 3D spheroids, and they have basically tested the per se effect of the renogrit on the whether it is toxic to the cells or not. So it is basically not toxic, and vancomycin, it basically produce toxicity in a concentration dependent manner. The 2 millimolar concentration of vancomycin was basically used to assess the effect of renogrit on vancomycin induced cytotoxicity. So as you can very well see over here, that renogrit significantly reduced the toxicity induced by vancomycin at a dose of 2 millimolar. Additionally, it could also reduce the levels of kidney injury markers like KIM1 and NAC in the HK2 spheroids. Finally, it also decreased the mRNA expression of NGAL and MMP7, which are also important kidney toxicity markers. So we also did a PGP efflux transport analysis. As we all know, PGP is the P-glycoprotein. It basically throws out whatever is thrown in the cell. Here we have used a substrate, calcine AM, which is a substrate for PGP, and as soon as it enters the cell, it undergoes hydrolysis to produce calcine, which fluoresces. Vancomycin inhibits this PGP, and it leads to greater fluorescence because calcine AM cannot be expelled out of the cell. So we measured the intraspheroidal fluorescence, and we could see that renogrit could, it basically restored the PGP mediated transport. So coming to the in vivo model, this is the entire model. I am not going in great details over here. Vancomycin, when given for 7 consecutive days, and we have given 12 doses, it led to the increase in the level of urea, creatinine, and these elevated urea and creatinine levels were dose-dependently decreased by renogrit. Similarly, renogrit improved the bun clearance and creatinine clearance from the blood in a concentration, in a dose-dependent fashion. And we have some derived parameters, and these are the levels of the urea nitrogen. It had gone down, it had gone up in the plasma, and it has gone down in the urine, and renogrit could restore the urea in the urine as well as in creatinine. It could also increase the estimated glomerular filtration rate in a dose-dependent manner. Renogrit, after giving vancomycin, what we observed was there was a relative increase in the weight of the kidney, which was brought down significantly by renogrit at different doses in a dose-dependent manner. The oxidative stress had also gone up in the kidneys, which was basically restored by the drug in a concentration-dependent manner. And these kidney injury markers were also brought down significantly with renogrit in a dose-dependent fashion. Additionally, renogrit also decreased the expression of kidney injury markers like KIP1 and osteopontin. And these are the beautiful histopathology photos where you can see cell death occurring over here, dilatation of the tubules, and treatment with various doses of renogrit ameliorated the tubular necrosis, tubular dilatation, cast cells, and inflammatory cell infiltration. So this is basically what is mentioned in the photos I have shown in the graphs over here. And we gave renogrit for 7 days. It did not alter any of the liver enzymes, and it did not alter any of the electrolytes. And that basically sums up that renogrit is basically safe when given for at least 21 days. So summary and conclusions are beautifully explained in this graph, wherein in vitro, the findings I have already discussed, and these are the in vivo findings. Hence, Renograte holds a clinico-therapeutic potential for iatrogenic kidney diseases, the kidney diseases caused by drugs, and the next plan is to assess the non-clinical safety evaluation of Renograte. Thank you so much.
Sandeep, very good talk. And it's very nice at the fastest way you delivered so much information. So thanks a lot. Guys, let's clap for the Sandeep, the last speaker for the day, and the way he hold the crowd was very marvelous. Thank you.
Thank you very much, sir. Trictions, Dr. Swati Haldar and Dr. Rakesh Maurya ji will give their talks tomorrow so that we can make the best out of it and enrich ourselves from their experiences. Also, we have moved today's panel discussion to day 3 tomorrow, as part of the topic was addressed by the experts yesterday, and the remaining will be covered tomorrow after the speaker's session. May I now request Professor Chintamani sir to kindly give closing remarks to summarize today's session. Let's all welcome Professor Chintamani sir.
Good evening. Good evening, everyone. First of all, a huge round of applause for all of you for sitting through the day. And I sincerely hope it made sense, because I personally thoroughly enjoyed it. Now, it is impossible to summarize the day, and all I'm going to do is share as I understood the whole day, and I'm sure you would chip in with your questions if there are any. Maybe you can have a couple of questions, because we are running terribly short on time. So I just have a few things to say which I understand from today's wonderful talks. We started the day with the divine talks from both Swamiji, Param Shraddheya Swamiji, and Acharya Shri. So huge round of applause for both of them for making it extremely special for us and blessing us to start the day on a good note. Dr. Anurag made it very, very, I mean, it's needless to say the kind of effort that goes into organizing such events, but a huge round of applause for Dr. Anurag for making it happen. And as far as these speakers for the day are concerned, each one was outstanding, and each one had so much to contribute to our knowledge, our learning. And you can clap for them even louder. Thank you very much. So I would not be individually summarizing each talk. I'll just take, as they say, the milk is from various cows, but the cheese is mine. So I'll give you at the end the summary of the whole thing. So let's start with the Martin's principle only. Nothing is difficult as long as someone else has to do it. And that is what was brought out very nicely in a couple of talks. Extremely difficult jobs that they do. And you must have realized there is one thing common in the whole days. I hope I can have your attention. It'll take just 10 minutes. Are you all right to listen to me? Yes, sir. A louder yes, please. Yes, sir. Even louder. Yes, sir. Thank you. So, nothing is difficult as long as someone else is doing it. So that I wanted to tell you specifically, because you were to sit through all the talks without knowing everything about it was a tough job, but you made the most of it. And Acharya Shri was saying, look at my students. We told them to sit, and all of them sat throughout the day. I truly would like to clap for you. As a teacher, it's very heartening to see students learning throughout the day, and I'm sure It's not just about information all the time. Please understand this. When we are sharing information, more importantly, we are sharing the ignorance. There's a lot that we don't know. And I don't know is a tremendous possibility. We don't know so many things. There's very little we know. There was a lot of talk on Alzheimer, the functioning of the brain. And we all know today, it's scientifically proven that we don't know more than 20% of our brain. So imagine 80% is still a possibility, and what all can be done. And that's what these dimensions are all about. And yoga brings it to your doorstep. And that's where you actually open up to these tremendous possibilities. There is a beautiful saying, in science especially, always is always wrong, and never is never right. So if anybody says, always do it this way, never listen to him. He's wrong. What was an apostle 20 years ago, that radical surgery cures cancer, only radical surgery, is gone. We now talk about organ preservation. I'm just giving an example. So many of these paradigms have shifted. What does that mean? It is going to change tomorrow also. So keep that possibility in your mind. So it's not the last time you're hearing about such wonderful ideas. They keep cropping up because we evolve with time. If anybody thinks he has learnt it all, he has learnt nothing. So basically, we just continue to share our ignorance and our information. I personally learned so much about it, especially towards the end of it. The talks made such an impact. There is so much we can do. And there is such a huge amount of potential if we get together. The bottom line is, in everybody's talk, if I stand up and say, surgery is the top part of it, we have left it long ago. I mean, gone are the days when he was considered tiger in the OT. You will take out anything except a tumor which is fixed to the operating table, it will all be removed. And my boss would teach me, surgeons don't listen to anesthetists or physicians or anybody else. They just take it out. But that's wrong. It's changed. And why is it changed? Because patient is coming in the center. If my patient is benefited by Pidonil or any other medicine, I would prefer to use it regardless of that stupid ego that a doctor may have. The reverse is also true. Don't go beyond a point explaining that we can cure everything. You can't. Nobody can. But you can contribute to the healing of the patient. Remember one more thing. No drug heals anybody. Individuals heal themselves. The best that any medication or treatment can do is help you heal yourself. So eventually, it's you who's doing it. And where is this background coming from? It is coming from our own very scriptures. And yoga is the oldest way of the way you should live so that you don't fall ill at all because you're healing yourself. The entire mechanism of pain management, apoptosis, we are learning it today. It was already happening in this body. What does it mean? It was always there. We are just discovering a bit of it. So whenever you give something from outside, unless it's a part of you, it will be rejected. Even if it cures one problem, it will get another. 50% of Americans are on at least one drug per day. And about 40% on 2 drugs per day. And these are popping pills like painkiller across the counter. Kidneys are getting damaged and rogered. And if you look at it, the blood pressure, high blood pressure was defined as 160 by 110 once upon a time, got down to 120 by 80. It keeps changing. Why is it changing? We are reducing it every day so that you go to a doctor, get treated. I don't want to leak out more secrets about us. There was one beautiful referral to butterfly PT, and I started Googling it. I wanted to understand this relating to Alzheimer. Well, Alzheimer, again, is preventable. And can you guess what prevents it? Brain lives on 2 things only. Oxygen and glucose. Brain needs nothing else. Everything else is required, but majority is this. That's why there was a myth earlier The vegetarians are more sharp than the non-vegetarians, the kind of food you eat. For a different reason, vegetarian food is always better because that's a human food. The way the animal is slaughtered, there's a study that has been conducted. Just before getting slaughtered, the animal has a very high content of adrenaline. If you are to be slaughtered, the content of adrenaline will rise. The metabolic products are carcinogenic. The cancer rates are higher in non-vegetarians, and nobody is surprised. This is not a rocket science. If you eat what you're meant to eat, you would not fall ill. What you eat, you become. Jaisa an, vaisa man, as it has been said in our scriptures. I have told you nothing, it's already known. It's remarkable that Shraddheya Acharya ji was talking about 52,000 species of plants that he has accumulated. He's bringing it out in the form of a granth, where actually Ayurveda would be rediscovered and actually retaught. There is a phenomenon in life and in life cycle, or the cycle of earth. You have to undo to redo, to undo again. So in the process we are evolving. I think that is a huge, huge amount of contribution to mankind, I must say. And this kind of a work can change the future of medicine. There can be a situation where you may require no medicine. Well, very interesting talk on nanoparticles. And nano means small, and as small as it can get. Do you know, somebody asked a question last time when I was here, do you really believe in God, sir? I was walking out, he wanted a selfie, and then he asked this question. I said, you don't? Have you seen an electron? No. You believe in electron? Yes. So, what's the problem? We have evolved all our theories, all our analysis based on the existence of neutron, proton, electron, before having seen them. Seeing is not believing. You see nothing. What you see is only 10% of what is existing. So that doesn't mean that 90% does not exist. If there is creation, there is a creator. When you appreciate a painting, you're appreciating a painter. Same concept applies to we as energy. And energy is what got transformed into matter. I was giving a reference of Ashtavakra where he's asking how does Shrishti come when he's having a Shastrat with Bandi. And that was a famous Shastrat where he says, well, it emerged like a spider throwing out the saliva, creating the web. That's the Shrishti, and then it swallows it, and that's the end of it. So in an ocean, the waves come up and go down. Ocean remains. That's the concept of our existence. So since we are just mere energy particles, we are all together and we are all in it. Basically, the idea is, I'm getting back to something that I've spoken in the morning. It is that cell you should focus on. If that cell is joyful, you are joyful. If you're joyful, joy is not happiness. Don't confuse the two. Joy is spiritual, happiness is physical. You can induce happiness, it will be short-lived. Joy is within, like you get after yoga, or after meditation, or praying, or doing something good. Why should you feel good after doing something good? Because that's the joy you're looking for. Each cell is looking for. And where does it all happen? Where do you fall sick and where do you heal? Mind. And where is the mind? Not here. All over the body. They found the same particles, the memory cells in the pancreas. So each cell has a memory. It has got a chip. It knows its ancestors. So don't fool the cell or the cell will fool you. So end of the day, all I would request and summarize the tremendous amount of talks. I was talking about propolis and beeswax, Dr. Purulia, we use regularly in commandos. When the mandible we resect, the bleeding happens. You can't cauterize it. So we pack the hell out of it using beeswax, which is bone wax. And I was asking Dr. Anurag, he said, but it's too thick. So it may not reach where you want it to reach, which is very tiny area. Making it into a nano, you can reach almost anywhere. But killing all bacteria is not great news. There are lots of friends around in our body. Like I said, the first one to appear was the germ. They share your aura. when people like Acharya Shri or Swamiji, they walk in, you say there's an aura. The aura is the same vibrations that you see around. They are actually that flora of bacteria, or the germs you call them. They are friends. If you have one cell, then 9 are that germs. So who is in majority? You are actually mostly germs. Only 1/10 of the whole scenario, the whole jiggery pokery, you are only 1/10. The rest is germs. And you kill them all, like washing with these chemicals that are available. I was really fascinated by the oral mouthwash, the Listerine mouthwash, which we have been using for ages for our oral cancer patients. It really burns the hell out of them. They feel terrible. And then they came out with this Listerine wash. One more, they added something to it. It was even worse. They stopped it. Now, this would be something I'll try my hands on, because 1, it is harmless. 2, it is going to work. Might as well do that. I can go on and on. That's a problem, because there's so much to speak. And I just have to condense it in one last sentence. That is, if you put the patient in the center and all of us around him, you know your job. If you put yourself in the center and the whole of the world around you, like I said, I decide what is to be done to the patient. I leads to, what is illness? I, illness. We, wellness. So work towards wellness. Thank you very much. Thank you.
Thank you very much, sir, for your encouraging words. अब मैं परम श्रद्धेय श्री आचार्य जी महाराज से सादर निवेदन करती हूँ कि वे आज के सभी स्पीकर्स को मोमेंटो प्रदान करें. With this, may I now call upon stage Professor Domenico Delfino, Professor Abhishek Parolia, Professor Chintamani sir, Professor Shubhini A. Saraf, Dr. Rakesh K. Singh, Dr. Kuldeep Singh, Dr. Kunal Bhattacharya, Mr. Sandeep Sinha, Thank you very much, everyone. Time to conclude day 2 of this conference, and let's end this day with the yoga dance performance by the students of University of Patanjali, so that we can bounce back tomorrow with same energy and level up our science. So we'll meet tomorrow at 9 a.m. again. And before we conclude the day, let's enjoy the yoga performance by the students from University of Patanjali. Let's give them a huge round of applause.
Volume, please.